Imagine a universe on the brink of unraveling where a single shift in vacuum energy could shatter reality. And the slow, inevitable heat death of the cosmos looms like an inescapable curse. In this video, we expose 25 jaw-dropping discoveries from the mind-bending power of CRISPR and synthetic biology that could resurrect extinct species, to melting polar ice and permafrost thawing that signal a world in collapse. Brace yourself as we dive into the dark underbelly of modern science, from self-replicating robots and memory manipulation to the unsettling possibility of mind uploading. I'd love to invite you into our cozy corner of the internet. Drop a quick comment and let us know where are you watching this from and what time is it for you. It's pretty cool how we're all connected, searching for that perfect night's sleep. If these scientific discoveries light up something in you, go ahead and subscribe our channel Restwise. We're more than just another sleep channel, we're a group of sleep explorers who know that a great night's sleep can change everything. Now, get comfy and let these mind-bending scientific discoveries guide you into a peaceful sleep. Vacuum decay, the ultimate cosmic doomsday. Imagine a bubble of absolute destruction, expanding at the speed of light. A wave of annihilation that rewrites the fundamental laws of physics, transforming our universe into something utterly alien. Something that would instantly erase all known matter, all stars, all planets, and all life. This isn't science fiction. This is vacuum decay, perhaps the most terrifying scientific concept ever discovered. The story begins with the Higgs field, quantum field permeating all of space that gives particles their mass. In 2012, scientists at the Large Hadron Collider confirmed the existence of the Higgs boson, the particle manifestation of this field. While this Nobel Prize-winning discovery completed the standard model of particle physics, it also revealed something deeply unsettling. According to our best calculations, our universe exists in what physicists call a false vacuum state. Think of it as a ball resting in a small depression on a hillside rather than at the bottom of the valley. It's stable for now, but it's not in its lowest possible energy configuration. There exists a true vacuum state with dramatically lower energy, and our universe might someday quantum tunnel into it. This is where the horror begins. If the Higgs field were to tunnel to its true vacuum state, somewhere in the cosmos, it would create a bubble of true vacuum that would expand outward at the speed of light. Inside this bubble, the laws of physics would be radically different. The fundamental forces, the masses of particles, everything would change in ways that would make matter as we know it impossible. The most disturbing aspect, we wouldn't see it coming. Since the bubble expands at light speed, no warning could outrace it. One moment, existence as normal, the next, oblivion. Not even death in the conventional sense. Just a fundamental transformation, rendering our form of matter impossible. The theoretical physicists who first extensively explored this concept, Sidney Coleman and Frank D. Loacher, described it in their 1980 paper with clinical detachment. The possibility that we are living in a false vacuum has never been a cheering one to contemplate. This masterpiece of scientific understatement belies the existential terror of their mathematics. How likely is vacuum decay? That depends on the precise mass of the Higgs boson and the top quark. When measured at the Large Hadron Collider, the Higgs mass came in at approximately 125 GeV gigaelectron volts, troublingly close to the danger zone. As Nobel laureate Joseph Incandela noted after the Higgs discovery, we're on the edge of stability. Later calculations by cosmologists like Andreas Moss at the University of Cambridge suggested our universe might remain metastable for billions or even trillions of years. But metastable is not stable. Our universe is like a pencil balanced on its tip. Given enough time, quantum fluctuations make decay inevitable. What's even more disturbing is that certain cosmic events might trigger vacuum decay. High-energy cosmic phenomena, black hole evaporation, or even advanced particle accelerators could theoretically provide the push needed to nucleate a true vacuum bubble. each time scientists push to higher energies in accelerators. Some worry, although most physicists consider it extremely unlikely, that we might accidentally trigger the end of everything. The calculations by theoretical physicist Thomas Hertog at KU Leuven suggest that extremely dense objects like neutron stars might be cosmic vacuum decay factories, environments where quantum tunneling becomes exponentially more likely. Every neutron star in the universe could be a loaded gun pressed against the temple of existence. In a macabre twist, some physicists find comfort in the very horror of vacuum decay, as theoretical physicist Katie Mack notes in her exploration of cosmic doom scenarios. If vacuum decay has already happened somewhere in the cosmos, and a bubble of true vacuum is heading our way, there's absolutely nothing we can do about it. The oblivion would be instantaneous and total. No suffering, no awareness. Simply a reconfiguration of reality at the most fundamental level. Perhaps most unsettling is that vacuum decay connects to the multiverse hypothesis. In the eternal inflation model proposed by cosmologist Alan Guth, vacuum decay might be happening constantly throughout a much larger multiverse, with bubbles of different vacuum states, creating entirely different universes with different physical laws. Our universe might be just one bubble in a cosmic foam of reality, temporarily stable but ultimately doomed. Stephen Hawking, in some of his final papers, proposed that vacuum decay might actually be necessary in the grand scheme of cosmic evolution. That universes must die for new ones to be born with different properties. Our doomed universe might be a necessary sacrifice in the grand experiment of reality. So we exist, balanced on a knife's edge of quantum probability. Every moment that passes without the nucleation of a vacuum decay bubble is, in a sense, beating the cosmic odds. We are living in a universe that should not exist for long by its own rules. Temporary bubble of borrowed time in a reality that mathematics suggests should have destroyed itself long ago. The ultimate cosmic horror isn't aliens, black holes, or even the heat death of the universe. It's that existence itself might be fundamentally unstable. That the ultimate scientific truth of our universe is that it contains the seeds of its own instantaneous and total destruction. A catastrophe that could begin at any moment, anywhere in the cosmos, without warning. The heat death of the universe. In the grand cosmic theater, few scientific discoveries evoke such profound existential dread as the heat death of the universe. The ultimate fate predicted by thermodynamics, where all energy becomes irretrievably dispersed, all motion ceases, and time itself loses meaning. The story begins in the 19th century with Rudolf Clausius and Lord Kelvin, who formulated the laws of thermodynamics. The second law, perhaps the most unyielding law in physics, states that entropy, a measure of disorder, always increases in a closed system. While seemingly innocuous, this principle carries catastrophic implications when applied to our universe as a whole. Unlike most cosmic catastrophes, heat death isn't violent. There are no explosions, no dramatic collapses. Instead, it's the quiet suffocation of possibility. Imagine the universe as an immense cosmic clock, wound tight at the Big Bang, gradually unwinding with each passing yawn, inevitably running down to perfect stillness. Today, our universe teems with activity. Stars fuse hydrogen into heavier elements, releasing the energy that makes life possible. Galaxies spin in magnificent spirals. Black holes devour matter, emitting radiation in their final moments. But these processes all rely on energy gradients, differences in temperature and concentration that allow work to be performed. As entropy increases, these gradients diminish, fast, forward 100 trillion years. The last stars will have burned out, their remnants cooling to the background temperature. Protons, once thought eternal, may decay after 10–40 years, leaving only a diffuse soup of photons, electrons, positrons, and neutrinos. Black holes, those voracious cosmic engines, will evaporate through Hawking radiation after 10–100 years. Even the fabric of space itself might eventually decay. Physicist Brian Greene describes this endgame as vast, empty, dark, and cold universe, where the universe reaches a state of maximum entropy, a state of equilibrium, where nothing happens and nothing changes. The universe becomes, in effect, a perfectly uniform, motionless void, a cosmos of absolute sameness. What makes heat death so profoundly disturbing is its inevitability. As theoretical physicist Lawrence Krauss notes, all the energy available within the observable horizon of the universe today will eventually be used up. No technology, no matter how advanced, can reverse entropy's inexorable march. Unlike other existential threats, this one is written into the fundamental equations of physics. The concept raises profound questions about meaning in a universe with a finite lifespan. Philosopher Bertrand Russell famously lamented, "All the labors of the ages, all the devotion, all the inspiration, all the noonday brightness of human genius are destined to extinction." If everything ultimately dissolves into the same homogeneous soup, what ultimate purpose can any action serve? Yet some scientists see nuance in this bleak picture. Theoretical physicist Roger Penrose proposed his conformal cyclic cosmology, suggesting that after heat death, the universe might become mathematically equivalent to another Big Bang, creating a new universe with its own arrow of time. Others point to quantum fluctuations that might, given the infinite time available after heat death, produce new universes through random chance. The mathematics of heat death also connects to information theory. As physicist Seth Lloyd demonstrated, all physical processes can be viewed as computations, manipulations of information. In this framework, heat death represents the state where no further information processing is possible. The universe's final thought, even more mind-bending, is the temporal perspective. As entropy maximizes, the distinction between past and future blurs. Physicist Julian Barber argues that in a state of heat death, time disappears completely. There is no difference between up and down, backward and forward. Our most fundamental experience, the passage of time, becomes meaningless. Recent observations of dark energy, which accelerates the universe's expansion, have only reinforced heat death's likelihood. As galaxies recede beyond our cosmic horizon, each pocket of the universe becomes increasingly isolated, hastening local heat death. Cosmologist Katie Mack explains, dark energy is essentially putting the universe on a fast track to a more desolate future. What's perhaps most unsettling is that we live in the brief cosmic moment when the universe is hospitable to complexity. As theoretical physicist Freeman Dyson calculated, the window for complex structures like stars, planets, and life is vanishingly small compared to the universe's total timeline. We exist in what cosmologists call the stelliferous era, a cosmological eye blink before eternal darkness. The heat death reveals a universe that is, in physicist Arthur Eddington's words, slowly and inexorably dying. Every structure we see, from galaxies to civilizations to consciousness itself, represents a temporary eddy in the cosmic current flowing toward perfect stillness. We build our lives in the shadow of entropy's final victory, a scientific truth that frames every human endeavor against the backdrop of ultimate cosmic futility. Yet in this bleakest of scientific discoveries lies a strange beauty. The recognition that complexity, consciousness, and meaning exist not despite the universe's mortality, but because of it. The brief flicker of order that permits our existence becomes all the more precious against the infinite darkness that awaits. The Heliocentric Revolution, for thousands of years, humanity stood confidently at the center of creation. The Earth, unmoving and special, formed the fixed stage upon which the cosmic drama unfolded. The sun, moon, planets, and stars all revolved around us in their perfect celestial spheres. Or so we thought. The Heliocentric Revolution didn't just rearrange the solar system. It fundamentally altered humanity's place in the cosmos and challenged our most basic perceptions of reality. The story begins not with Copernicus, as commonly believed, but with ancient astronomers who first questioned Earth's central position. Around 250 BCE, Aristarchus of Samos proposed that the Earth rotated daily and orbited the Sun annually. His heliocentric model was largely rejected, overshadowed by Aristotle's geocentric system and later formalized by Ptolemy in the 7th century CE. For nearly 1400 years afterward, this Earth-centered model dominated Western and Islamic astronomy. The architecture of this geocentric universe was both elegant and reassuring. Earth sat motionless at the center, surrounded by crystalline spheres carrying the Moon, Sun, planets, and stars in perfect circular orbits. This model aligned neatly with Aristotelian physics, which taught that heavy objects naturally move toward the center of the universe. Earth, while celestial bodies, made of perfect quintessence, moved naturally in circles. Yet problems plagued this elegant system. The observed retrograde motion of planets, where they appear to move backward in the sky, required increasingly complex explanations. Ptolemy introduced epicycles, circles upon circles, to account for these irregularities. By the Middle Ages, the model had become unwieldy, requiring dozens of epicycles to match observations. Enter Nicholas Copernicus, a Polish canon and astronomer who would fundamentally reshape our cosmic understanding. Working quietly for decades, Copernicus developed a comprehensive heliocentric model, placing the Sun at the center with Earth as just another planet. He delayed publishing his revolutionary De Revolutionibus Orbium Coelestium until 1543, the year of his death, perhaps aware of the controversy it would spark. Copernicus's model wasn't immediately more accurate than Ptolemy's. He still insisted on circular orbits and needed epicycles. But it was mathematically simpler and more elegant. More importantly, it explained retrograde motion. Naturally, planets appear to move backward when Earth overtakes them in its orbit. The Danish astronomer Tycho Brahe, though skeptical of heliocentrism, unknowingly provided the data that would confirm it. For decades, he meticulously recorded planetary positions with unprecedented accuracy. After his death, these observations fell to his assistant, Johannes Kepler. Kepler, a brilliant mathematician, spent years analyzing Brahe's data, particularly Mars's orbit. After countless failed attempts, he made a crucial breakthrough. Planets move in ellipses, not circles, with the Sun at one focus. His 3 laws of planetary motion, published between 1609 and 1619, provided the mathematical framework for a workable heliocentric model. But the revolution's most dramatic chapter belonged to Galileo Galilei. When he pointed his newly improved telescope skyward in 1609, he made discoveries that devastated the geocentric worldview. He observed mountains and craters on the Moon, contradicting the belief in perfect celestial bodies. He discovered Jupiter's moons, proving that not all cosmic bodies orbited Earth. Most damning, he observed the phases of Venus, which could only be explained by the planet orbiting the Sun. Galileo's public advocacy for heliocentrism led to his infamous clash with the Catholic Church. In 1633, he was forced to recant his views and spent his remaining years under house arrest. Yet his observations had dealt the geocentric model a fatal blow. The revolution culminated with Isaac Newton's Principia in 1687. Newton's laws of motion and universal gravitation provided the physical explanation for Kepler's elliptical orbits. Gravity, the same force that makes apples fall, kept planets in their orbits around the Sun. The heliocentric model was now complete, both mathematically and physically. The implications extended far beyond astronomy. Philosophically, the heliocentric revolution triggered what historian Alexandre Coyer called the destruction of the cosmos. The collapse of the hierarchical, closed universe of ancient and medieval thought. Earth was demoted from cosmic center to ordinary planet. The universe expanded dramatically in both size and age. Theologically, the revolution challenged religious narratives that placed humanity at creation's focal point. If Earth was merely one planet among many, orbiting an ordinary star, what became of humanity's special relationship with the divine? As philosopher Blaise Pascal would later write, "The eternal silence of these infinite spaces frightens me." Methodologically, the revolution transformed how we acquire knowledge. The victory of the heliocentric model demonstrated the power of mathematics, observation, and experiment over tradition and intuitive perception. After all, the heliocentric model triumphed despite contradicting our daily experience. The ground feels stationary beneath our feet, not spinning at 1,000 miles per hour. Perhaps most unsettling was the psychological impact. As Freud would later note, Copernicus delivered the first great blow to humanity's narcissism. No longer could we see ourselves as occupying a privileged cosmic position. We were residents of a peripheral planet orbiting an average star in what we would eventually learn is one galaxy among billions. The Heliocentric Revolution thus stands as the first great scientific dethroning of humanity, a profound demonstration that the universe was not designed around us, that our intuitions about reality could be fundamentally wrong, and that the cosmos operated according to impersonal natural laws rather than human-centered purpose. In removing Earth from the center of the universe, it paved the way for a much larger, older, and more humbling cosmos than anyone had dared imagine. The Copernican Principle, when we gaze up at the night sky, an uncomfortable truth lurks behind the beauty of the stars. A scientific principle so profound and unsettling that it fundamentally altered our conception of our place in the universe. The Copernican Principle, named after the 16th century astronomer who dared to displace Earth from the center of creation, delivers a simple yet devastating message. We are not special. Our position in the cosmos is not privileged. We are, in cosmic terms, utterly ordinary. This principle emerged from Nicholas Copernicus's revolutionary heliocentric model, but its implications extend far beyond simply rearranging the planets. What began as an astronomical hypothesis has evolved into a cornerstone of modern cosmology, a methodological assumption that has repeatedly revealed the universe to be vaster, older, and more indifferent to human existence than anyone could have imagined. The Copernican Principle states that Earth occupies no privileged position in the universe. Initially, this meant only that Earth orbits the Sun rather than vice versa. But as our observational capabilities expanded, so too did the scope of our cosmic demotion. The Sun, once thought special, proved to be one of billions of stars in the Milky Way. Our galaxy, once thought unique, became one among billions of galaxies. Even our location within our galaxy turned out to be unremarkable, not at the center, but in the mundane suburbs of a spiral arm. In the 1920s, Edwin Hubble's observations of distant galaxies delivered another blow to human exceptionalism. The universe was not static but expanding, with galaxies rushing away from each other in all directions. This discovery eliminated any notion of a cosmic center altogether. As cosmologist Stephen Hawking wrote, "The Earth is a medium-sized planet orbiting around an average star in the outer suburbs of an ordinary spiral galaxy, which is itself only one of about a million galaxies in the observable universe." The principle found mathematical expression in the cosmological principle, which states that on sufficiently large scales, the universe is both homogeneous, the same in all places, and isotropic, the same in all directions. This principle underpins the standard model of cosmology and has been validated by observations of the cosmic microwave background radiation, the afterglow of the Big Bang itself. Astronomer Carl Sagan captured the existential weight of the Copernican Principle in his description of Earth as a pale blue dot in the vast cosmic ocean. Our posturings, our imagined self-importance, the delusion that we have some privileged position in the universe, are challenged by this point of pale light. The implications extend beyond our spatial position. Geologically, we learned that Earth is 4.5 billion years old, with humans occupying only the most recent fraction of that timeline. Biologically, Darwin's theory of evolution revealed humans to be just another species produced by natural selection, not specially created. Anthropologically, cultural studies showed that human societies develop according to common patterns, not divine plans. Even our consciousness, once thought uniquely human, has been shown to exist in varying degrees across many species. Perhaps most disturbing is the temporal extension of the Copernican Principle. Physicist Brandon Carter formalized this as the principle of mediocrity, suggesting we should not assume we live at a special time in the universe's history. Current evidence indicates we've arrived early in cosmic history. The era of stars and galaxies will continue for trillions of years, with potential for life long after our sun has burned out. The Copernican Principle even undermines our intuitive understanding of physics. We now know that the everyday physics we experience, the familiar forces, particles, and laws, represent just a thin slice of a much stranger reality. Dark matter and dark energy, which together compose 95% of the universe's content, remain mysterious to us. The quantum realm behaves in ways that defy our intuitions. Even time itself may be an emergent property, rather than a fundamental aspect of reality. Cosmologist Max Tegmark has proposed taking the Copernican Principle to its ultimate conclusion with his mathematical universe hypothesis, suggesting our universe may be just one among many in a vast multiverse, each with different physical laws and dimensions. If true, even our universe's physical constants aren't special, just the particular set that allows for our existence. The Copernican Principle has proven incredibly powerful as a scientific tool. Whenever we've assumed Earth or humans are special, we've been wrong. Whenever we've applied the principle, assuming mediocrity rather than privilege, we've uncovered deeper truths about reality. Yet this principle delivers what philosopher John Danaher calls a successful assault on human narcissism. Each application of the Copernican Principle has pushed humans further from the center of reality, transforming us from the crown of creation to cosmic accidents on an unremarkable planet orbiting an average star in one galaxy among billions. Perhaps the most terrifying aspect of the Copernican Principle is how it shapes our cosmic future. If we are not special, then the fate that has befallen countless species before us, extinction, likely awaits us as well. The principle suggests that civilizations like ours are probably common in the universe, yet the silence from the stars, the Fermi paradox, hints at a disturbing possibility. Technological civilizations may typically destroy themselves or face extinction before achieving long-term sustainability. The astronomer J. Richard Gott applied the Copernican Principle to human longevity and calculated that, with 95% confidence, humanity will survive between 100 and 7.8 million more years. A wide range, but both bounds are disturbingly finite. In stripping away our cosmic significance, the Copernican Principle forces us to confront the possibility that we exist in a universe that neither knows nor cares about us. That our hopes, fears, achievements, and civilizations may be, in cosmic terms, as significant as bacteria growing on a rock. This is not merely an astronomical discovery, but an existential one. A scientific principle that reaches beyond telescopes and equations to touch the very core of what it means to be human in an indifferent universe. Galileo's telescopic discoveries, in the winter of 1609, a 45-year-old Italian mathematician pointed a crude instrument toward the heavens and shattered humanity's conception of the cosmos. Galileo Galilei didn't invent the telescope. Dutch spectacle makers had created simple spyglasses a year earlier, but he was the first to systematically turn this new technology skyward. What he observed through his homemade device, capable of only 20x magnification, would devastate centuries of established wisdom and forever alter our place in the universe. The scientific establishment of Galileo's time subscribed to an Aristotelian, Ptolemaic cosmology, a perfect, immutable universe with Earth fixed at its center. The heavens were thought to be perfect and unchanging, composed of crystalline spheres, carrying the celestial bodies in perfect circular motion. This cosmic architecture wasn't merely astronomical, it was a comprehensive worldview binding together physics, philosophy, and theology. Galileo's observations methodically dismantled this entire cosmic framework. His first target was the Moon, which he studied meticulously in November and December of 1609. Through his telescope, the Moon revealed a landscape shocking in its familiarity. Mountains casting shadows, deep craters, and vast plains he called Maria, as seas. In his revolutionary work, Sidereus Nuncius, so the Starry Messenger, published in March 1610, Galileo wrote it is like the face of the earth itself, certainly not polished to mathematical regularity, but uneven, rough, and full of cavities and prominences, not unlike the valleys and mountains covering the earth's surface. This observation delivered a profound blow to Aristotelian cosmology. If the moon, a celestial body, bore terrestrial features like mountains and valleys, then the fundamental distinction between the perfect heavens and corrupt earth collapsed. The cosmos suddenly appeared more uniform, governed by the same physical principles throughout. But Galileo was just beginning. Turning his telescope toward Jupiter in January 1610, he noticed 3 small stars, arranged in a straight line near the planet. Night after night, he observed these points of light changing position yet remaining near Jupiter. A 4th star soon appeared. Galileo realized these weren't stars at all, but moons orbiting Jupiter. The first celestial bodies observed to orbit something other than Earth. The implications were revolutionary. If moons could orbit Jupiter, then clearly not everything in the heavens revolved around Earth. The Jupiter system provided a miniature model of the Copernican solar system, with Jupiter representing the Sun and its moons the planets. As Galileo noted, this discovery alone removes the principal objections to the Copernican system. When Galileo pointed his telescope at the Milky Way, another shock awaited. What appeared to the naked eye as a cloudy band resolved into countless individual stars. Thousands upon thousands, too numerous and faint to be seen individually without telescopic aid. The universe suddenly expanded exponentially, populated with far more celestial bodies than anyone had imagined. Perhaps his most devastating observation came when he examined Venus. He discovered that Venus, like the moon, displayed phases. More crucially, Venus exhibited a full set of phases, from thin crescent to full disk, with its apparent size changing dramatically throughout. This observation was incompatible with the Ptolemaic system, where Venus could never appear full when viewed from Earth, but perfectly aligned with the Copernican model. His observations of Saturn confused him. His primitive telescope couldn't clearly resolve Saturn's rings, making the planet appear to have strange handles or companions that seemed to disappear and reappear over time. Similarly, his observations of sunspots, dark blemishes on the solar surface that formed, moved, and disappeared over days or weeks, further undermined the Aristotelian notion of perfect, unchanging celestial bodies. These discoveries, published in Sidereus Nuncius and subsequent works, ignited a scientific revolution. The implications reached far beyond astronomy. If the heavens weren't perfect and unchanging, if Earth was just another planet, if Jupiter had its own system of moons, then the entire medieval worldview, with its neat hierarchical cosmos reflecting divine order, crumbled. The religious authorities recognized the danger. In 1616, the Catholic Church placed Copernicus's work on its index of prohibited books and ordered Galileo to abandon the heliocentric view. When Galileo later published his Dialogue Concerning the Two Chief World Systems in 1632, clearly favoring the Copernican system, he was summoned to Rome, tried by the Inquisition, and forced to recant. Legend claims that after his recantation, Galileo muttered, "A per se move," and yet it moves. Though this story likely originated later, the philosophical implications of Galileo's discoveries were profound. His observations suggested that the universe was governed by mathematical laws, discoverable through systematic observation and experiment, not by appeals to ancient authority. As he famously wrote, "The book of nature is written in the language of mathematics." Galileo's methodological approach, combining systematic observation, experiment, and mathematical analysis, established the foundation of modern science. He insisted that sensory evidence, properly obtained and analyzed, trumped traditional authority. When critics refused to look through his telescope to see Jupiter's moons for themselves, Galileo recognized that he faced not just scientific disagreement, but a clash between two irreconcilable approaches to knowledge. What made Galileo's telescopic discoveries so terrifying was not just their contradiction of established theory, but their accessibility. Anyone with a telescope could verify them. No specialized knowledge or mathematical training was required to see Jupiter's moons or the phases of Venus. The evidence was democratized, visible to any who would simply look. In shattering the crystalline spheres of the ancient cosmos, Galileo did more than advance astronomy. He fundamentally altered humanity's conception of its place in the universe and our relationship to knowledge itself. His telescopic discoveries revealed that the cosmos was not created with humans at its center, that scientific truth could contradict both intuition and authority, and that the universe operated according to natural laws indifferent to human preference. With a simple tube containing two pieces of glass, Galileo exposed the universe as vaster, older, and less human-centered than anyone had dared imagine, a scientific revelation as terrifying as it was profound. Expanding universe, a cosmic revelation of endless retreat. For thousands of years, humans assumed the universe was static, a vast, unchanging stage upon which the cosmic drama played out. Stars might move across the sky, planets might wander between constellations, but the fundamental architecture of the cosmos was presumed eternal and unchanging. The discovery that our universe is actually expanding, and has been for billions of years, ranks among humanity's most profound and unsettling scientific revelations. The story begins in the early 20th century with a Belgian priest and physicist named Georges Lemaitre. In 1927, Lemaitre published a groundbreaking paper that proposed an expanding universe based on Einstein's general relativity equations. He suggested that the universe originated from what he called a primeval atom, or cosmic egg, the first theoretical conception of what would later be known as the Big Bang. Yet Lemaitre's work initially received little attention. 2 years earlier, American astronomer Edwin Hubble had made a monumental discovery using the 100-inch Hooker telescope at Mount Wilson Observatory. He proved conclusively that the Andromeda Nebula was actually a separate galaxy far beyond our Milky Way, instantly expanding the known universe by orders of magnitude. The universe wasn't just our galaxy, it contained countless other island universes scattered throughout the void. But Hubble's most profound discovery came in 1929. Building on earlier work by Vesto Slipher, who had measured the Doppler shifts of galactic light spectra, Hubble plotted the distances of galaxies against their redshifts. The stretching of light toward the red end of the spectrum that indicates objects moving away from the observer. The pattern that emerged was astonishing. Nearly all galaxies were receding from us, and the farther away they were, the faster they moved. This relationship, now known as Hubble's law, led to the inescapable conclusion that the universe itself was expanding. As Hubble wrote with characteristic understatement, the data in the table may be represented by a linear relation between distances and velocities. This seemingly modest claim concealed its revolutionary import. The universe had a history, a beginning, and was undergoing continuous, dramatic change. The expanding universe presented an immediate challenge to Einstein himself. When Einstein first applied his general relativity equations to the cosmos, they predicted an unstable universe that would either expand or contract. Finding this idea aesthetically displeasing, Einstein added a cosmological constant to his equations to force a static universe. Upon hearing of Hubble's discovery, Einstein reportedly called the cosmological constant his greatest blunder. The implications were profound. If the universe was expanding, then rewinding the cosmic film suggested a moment of creation, a beginning to space and time themselves. The idea was so radical that many scientists, including Fred Hoyle, proposed alternative, steady-state theories to avoid the philosophical and theological implications of a cosmic beginning. Hoyle mockingly coined the term Big Bang to ridicule the idea, never imagining the name would stick. But the evidence continued to mount. In 1964, radio astronomers Arno Penzias and Robert Wilson detected a faint microwave signal coming from all directions in the sky. This cosmic microwave background radiation, predicted years earlier by George Gamow, Ralph Alpher, and Robert Herman, represented the afterglow of the Big Bang itself. The ancient light from when the universe first became transparent about 380,000 years after its birth. The expanding universe forces us to contemplate trulismic timescales. Current measurements place the age of the universe at approximately 13.8 billion years. The light from the most distant observable galaxies has been traveling toward us for over 13 billion years. When that light began its journey, Earth didn't exist. When dinosaurs roamed our planet, that ancient light was already billions of years into its journey. Perhaps most disturbing was the discovery in the 1990s that the universe's expansion is accelerating. Two independent teams led by Saul Perlmutter, Brian Schmidt, and Adam Riss studied distant supernovae and found that, contrary to expectations, the cosmic expansion wasn't slowing down due to gravity. It was speeding up. This acceleration pointed to a mysterious repulsive force, now called dark energy, which constitutes roughly 68% of the universe's total energy content. The accelerating expansion points to a bleak cosmic future. Galaxies beyond our local group will eventually recede faster than light due to the expanding space between us, disappearing from our observable universe forever. In the far future, beings evolving in the Milky Way would have no observational evidence that other galaxies ever existed. The evidence of cosmic expansion literally disappears over time. Physicist Lawrence Krauss describes this as the ultimate Copernican revolution. Not only are we not at the center of the universe, but we appear to exist during a privileged cosmic epoch, the only time when beings could observe both the universe's past, through the cosmic microwave background, and its vast scale, through distant galaxies. Future civilizations will inhabit an increasingly empty cosmos. The expanding universe concept has also given rise to even more mind-bending theories. Inflation theory, developed by Alan Guth and others, proposes that the universe underwent a period of exponentially rapid expansion in its first fraction of a second, explaining the remarkable uniformity of the cosmic microwave background. This theory further suggests our observable universe may be just one bubble in a much larger multiverse. Regions of space, time eternally inflating and giving birth to new universes with potentially different physical laws. What makes the expanding universe so profound is how it transforms our fundamental conception of reality. Space itself is not a fixed stage but a dynamic, evolving entity. The cosmos had a beginning and will undergo endless change. We exist at a particular moment in a vast cosmic story, witnessing a universe that emerged from conditions we can barely comprehend and heading toward a future of endless, accelerating dispersal. The expanding universe reveals that even emptiness itself, the vacuum of space, contains energy and undergoes evolution. It suggests that nothingness is unstable, that the natural state of the cosmos is not eternal stasis, but perpetual becoming. In this discovery, we confront not just the limits of our previous cosmic understanding, but the unsettling realization that the universe is fundamentally impermanent, caught in an eternal dance of creation and dissolution that dwarfs human existence in both scale and significance. For CRISPR and gene editing, in the shadow-filled halls of our scientific journey, few discoveries have cast as long and complex a shadow as CRISPR-Cas9. This revolutionary gene editing technology has been hailed as miraculous medicine, condemned as playing God, and feared as a potential harbinger of our own destruction. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, wasn't invented by humans at all. We merely discovered it. For billions of years, bacteria have been using this system as a primitive immune response against viral invaders, storing fragments of viral DNA like wanted posters to recognize future attacks. In 2012, scientists Jennifer Doudna and Emmanuel Charpentier realized this bacterial defense mechanism could be repurposed into something unprecedented, a precise genetic editing tool. What makes CRISPR truly nightmarish isn't what it is, but what it enables. For the first time in our history, We possess the power to directly edit the code of life itself with the relative ease of correcting a typo. Previous genetic engineering tools were like editing a document with scissors and glue. CRISPR is a word processor for DNA. The implications are staggering. CRISPR can potentially cure genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease by directly repairing mutated genes. In 2020, researchers successfully used CRISPR to edit cells inside a human body to treat a rare genetic blindness. But while we celebrate these medical miracles, we must confront the darker potential. In 2018, Chinese scientist He Jiankui shocked the world by announcing he had created the first CRISPR-edited babies, twin girls altered to resist HIV infection. This wasn't treating a disease, it was engineering human traits in embryos that would pass those changes to future generations. The global scientific community condemned this rogue experiment, but the genie was out of the bottle. Humans had officially entered the age of directed evolution. The most chilling aspect of CRISPR may be its accessibility. Unlike nuclear weapons or other technologies of mass destruction, CRISPR kits can be purchased online for a few $100s. This democratization of gene editing means the technology exists outside the realm of strict regulation. In 2019, a biohacker live-streamed himself attempting to modify his own DNA using CRISPR in his home laboratory. Then there's the specter of gene drives, a technique that could force engineered genes through entire wild populations. Imagine releasing mosquitoes edited to be incapable of carrying malaria, with the modification designed to spread through the entire species. Noble intention, but what of unintended consequences? The deliberate extinction or fundamental alteration of an entire species could trigger ecological collapse in ways we cannot predict. Military applications present perhaps the darkest possibilities. CRISPR could theoretically be used to create targeted bioweapons designed to affect people with specific genetic markers, ethnic bioweapons. While international laws prohibit such development, the technology exists. In 2016, U.S. Director of National Intelligence James Clapper added gene editing to the list of potential weapons of mass destruction and proliferation. Even more unsettling is the rise of genetic modification. In 2017, a research team led by Kevin Asvelt at MIT demonstrated how gene drives could potentially be used to alter wild mouse populations. While their research was conducted with safety protocols, it illustrated that a small team with modest resources could potentially alter an entire ecosystem. The ethical questions are as daunting as the technical ones. Who decides which genes are defects, to be eliminated versus natural human variation? Where is the line between treatment and enhancement? As CRISPR makes genetic modification more accessible, will we create new forms of inequality between the genetically enhanced and the unmodified? Consider the 2019 Russian biologist Denis Rubrikov's announcement of his intention to create more gene-edited babies, targeting a gene that causes deafness. While preventing deafness might seem uncontroversial, many in the deaf community view deafness not as a disability, but as a culture and identity. Who decides which human traits deserve elimination? As with nuclear fission before it, CRISPR presents humanity with godlike power before we've developed the godlike wisdom to wield it responsibly. The technology's co-inventor Jennifer Doudna has described having nightmares about Hitler asking to learn about her discovery. What makes CRISPR truly terrifying isn't just its power, but its inevitability. The knowledge cannot be unlearned. The technology cannot be uninvented. We stand at an evolutionary crossroads where, for the first time, a species has gained the ability to direct its own genetic destiny and that of every living thing around it. Perhaps most disturbing is the realization that we're just at the beginning. CRISPR is constantly being refined. In 2020, researchers developed prime editing, an even more precise form of CRISPR that reduces off-target effects. As our genetic editing tools become more sophisticated, the ethical guardrails become increasingly important. The question isn't whether humanity will rewrite the book of life, but how the story will unfold. Will CRISPR lead to a future where genetic disease is a memory, or to a dystopia of genetic castes and ecological devastation? The answer, frighteningly enough, rests in our still evolving hands. Synthetic biology. In laboratories across the world, scientists are no longer just studying life. They're redesigning it. Synthetic biology represents humanity's transition from passive observers of nature to active architects of living systems. While traditional genetic engineering modified existing organisms, synthetic biology aims to build life from scratch, following design principles more familiar to engineers than biologists. The genesis of this field traces back to 2000, when researchers first created synthetic biological circuits functioning like computer logic gates inside living cells. By 2010, Craig Venter and his team announced they had created synthetic life, a bacterium controlled entirely by a human-made genome. They called it Mycoplasma JCVI, SYN 1.0. But it represented something far more profound, the first organism in Earth's 4 billion-year history whose parent was a computer, not another living thing. What makes synthetic biology particularly unsettling is how it dissolves the boundary between the natural and artificial. When MIT scientists created bacteria that function as living cameras, storing images in their DNA, they demonstrated that living things could be reprogrammed as biological machines. The line between organism and technology began to blur. The power of synthetic biology comes from its modular approach. Scientists have developed bricks, standardized genetic components that can be combined like biological Lego pieces. These genetic building blocks form the foundation of what researchers call a biological internet, where genetic codes are shared, downloaded, and implemented globally. The registry of standard biological parts now contains thousands of these components, accessible to researchers worldwide. Perhaps most alarming are the military applications being explored. DARPA, the Pentagon's research arm, has invested heavily in synthetic biology through programs like Living Foundries and biological robustness in complex settings. Their stated goals are creating microbes that turn soil into concrete, like surfaces for landing strips, or developing organisms that detect explosives. The unstated potential, biological weapons of unprecedented specificity and lethality, lurks as an unspoken possibility. In 2017, researchers at the University of Alberta synthesized horsepox virus, a relative of smallpox, using mail-order DNA and publicly available information. Though not directly dangerous to humans, the experiment proved that even eradicated pathogens could be recreated from scratch. The team spent just $100,000 and completed the project in 6 months. The publication of their methods sparked fierce debate about whether such knowledge should be freely available. The commercial sector has embraced synthetic biology with equal fervor. Companies like Ginkgo Bioworks design custom microorganisms that produce everything from fragrances to food additives. Their facilities resemble software companies more than traditional laboratories, with automated systems designing and testing thousands of genetic combinations simultaneously. Their slogan, "Biology by Design," captures the essence of the synthetic biology revolution. The ecological implications are equally profound. Scientists have designed microalgae to produce biofuels at scales that could theoretically replace petroleum. Others have engineered bacteria to extract rare minerals from mine waste or to remediate environmental contamination. Yet releasing such organisms into the wild presents unprecedented risks. Even with built-in safeguards, synthetic organisms could transfer engineered genes to natural populations or evolve in unpredictable ways. In 2019, researchers at ETH Zurich created the first bacterial genome designed entirely by an algorithm. The computer rearranged the genetic code while preserving essential functions, creating a viable organism with a fundamentally alien genetic architecture. This computational life raises disturbing questions about our understanding of biology itself. Most chilling are the experiments in orthogonal biology, creating life that operates on principles entirely different from terrestrial biochemistry. Researchers have expanded the genetic alphabet beyond the natural 4 DNA bases, creating organisms with 6-letter genetic codes. Others have designed microbes that incorporate artificial amino acids not found in nature. These organisms represent a parallel biology, potentially incompatible with Earth's evolutionary history. The specter of do-it-yourself synthetic biology looms large as well. Community laboratories like BioCurious and GenSpace offer public access to many basic tools of the field. While promoting innovation, these spaces potentially democratize technologies of unprecedented power. In 2019, a California biohacker attempted to create genetically modified herpes viruses as a potential therapy, conducting experiments in his apartment laboratory. Particularly concerning is the accelerating pace of the field. The technology to sequence and synthesize DNA improves exponentially, following curves similar to computing power. The Human Genome Project took 13 years and cost nearly $3 billion. Today, a genome can be sequenced in hours for under $1,000. DNA synthesis follows similar trends, making the technology increasingly accessible to smaller labs and individuals. What separates synthetic biology from other technological revolutions is its self-propagating nature. Unlike computers or nuclear weapons, engineered organisms can reproduce, evolve, and spread autonomously. A mistake once released could replicate itself indefinitely. In 2020, mathematical modeling by researchers at MIT suggested that certain types of engineered gene drives could potentially spread globally from a single release, regardless of containment efforts. As we stand at this biological frontier, we face a stark realization. We have developed the power to rewrite the book of life before fully understanding what it says. Synthetic biology offers solutions to humanity's greatest challenges: disease, environmental degradation, resource scarcity, yet simultaneously presents risks of unprecedented magnitude. The technology to create new forms of life now exists, advancing faster than our capacity to predict its consequences or develop appropriate safeguards. In the words of synthetic biologist Drew Endy, "We're building things that have never existed before." Whether that represents humanity's greatest achievement or its most terrifying mistake remains to be seen. RNA World Hypothesis. Deep in Earth's primordial past lies a mystery that continues to haunt the scientific community. How did the extraordinary complexity of life arise from the sterile chemistry of the early planet? The RNA World Hypothesis offers an answer that is both elegant and unsettling, revealing a lost world where our most fundamental understanding of life itself must be reconsidered. The terrifying implication of the RNA world is not what it reveals about our past, but what it suggests about our fundamental nature. Life as we know it may be merely the second genesis, a successor system that overthrew its own creator. To understand this hypothesis, we must first confront the paradox that has long perplexed scientists. Modern cells rely on a seemingly impossible circular dependency. DNA stores genetic information but requires proteins to replicate it, while proteins carry out cellular functions but require DNA's instructions to be built. This biological chicken and egg problem points to a disturbing reality. Neither could have emerged first in isolation. Enter RNA, a molecular cousin to DNA that exhibits a disturbing duality. Like DNA, it can store genetic information, but unlike DNA, certain RNA molecules, called ribozymes, can also catalyze chemical reactions similar to proteins. This dual nature makes RNA a prime suspect for life's original molecule, both the blueprint and the builder. In 1982, Thomas Seck and Sidney Altman independently discovered the first natural ribozymes, RNA molecules that cut and splice other RNAs. This revolutionary finding earned them the 1989 Nobel Prize and provided the first tangible evidence that RNA could have served as life's original foundation. What they had uncovered was essentially a molecular fossil, a remnant of an ancient biological world that had been largely erased. More disturbing evidence exists within our own cells. The ribosome, the cellular factory that manufactures all proteins, is primarily composed of RNA, not protein, with a catalytic center that forms peptide bonds being entirely RNA-based. This suggests we still carry the architecture of our RNA ancestors within us, like biological ghosts haunting our cellular machinery. Laboratory experiments have shown just how plausible this ancient RNA dominance might have been. In 2001, researchers at the Scripps Research Institute developed ribozymes capable of catalyzing their own replication, demonstrating that self-replicating RNA systems could indeed have emerged spontaneously. By 2018, scientists at the MRC Laboratory of Molecular Biology had created ribozymes that could synthesize RNA strands up to 206 nucleotides long, nearly the size of some functional modern ribozymes. What's particularly chilling is how this primordial RNA world may have emerged. Experiments by Nobel laureate Jack Szostak and others have shown that simple fatty acids spontaneously form membrane-like structures under early Earth conditions. These protocells could have encapsulated replicating RNA molecules, creating primitive cells driven solely by RNA-based metabolism and replication. The frightening implications extend beyond Earth. If the RNA world represents a universal pattern rather than a historical accident, it suggests a specific pathway that life might follow on any suitable planet. The 2020 detection of phosphine in Venus's atmosphere, a potential biosignature, raised the disturbing possibility that we might find RNA-based life forms elsewhere in our own solar system, perhaps following evolutionary paths entirely different from our own. Evidence of this ancient RNA dominance persists throughout biology. Beyond ribosomes, small RNA molecules regulate gene expression in all organisms. The human genome itself is haunted by these ancient controllers. Approximately 98% of RNA transcribed from human DNA doesn't code for proteins, but instead performs regulatory functions. We are, in essence, still partially operating on RNA-based software that predates our current DNA protein operating system. Laboratory reconstructions of the RNA world have revealed just how alien this precursor form of life might have been. In 2015, researchers demonstrated that RNA can catalyze the formation of another RNA from smaller fragments without using any protein enzymes. This suggests an ancient biochemistry utterly different from what powers life today. A shadow biosphere that once dominated Earth, but has since been almost completely erased. Most disturbing is the story of transition. How did we move from an RNA world to our current DNA, protein world? The evidence points to something like an ancient coup d'etat. At some point, RNA molecules began catalyzing the synthesis of the first proteins, which proved more efficient at certain functions. DNA, which is more stable than RNA, eventually took over as the genetic storage medium. The RNA world was gradually overthrown by its own creations. This biochemical revolution left behind a compromised system. The complicated machinery of modern cells, with their interdependent DNA, RNA, and proteins, represents not an optimal design, but the scars of this ancient transition. We are built upon the remains of a prior biological paradigm, carrying within our cells the legacy of a life system almost entirely subsumed. Recent research has pushed the RNA world concept into even stranger territory. In 2016, researchers at the Scripps Research Institute created synthetic nucleotides that could be incorporated into RNA and DNA, expanding the genetic alphabet beyond the natural 4 letters. When these artificial bases were added to RNA molecules, some gained entirely new catalytic abilities, suggesting the RNA world could have been even more chemically diverse than previously imagined. The philosophical implications are perhaps most disturbing of all. If life began as self-replicating chemical systems that gradually increased in complexity, the boundary between chemistry and biology, between non-life and life, becomes disconcertingly blurred. The RNA World Hypothesis suggests life didn't appear in a dramatic moment of creation, but emerged through incremental increases in molecular complexity until self-sustaining chemical systems crossed an ill-defined threshold into what we recognize as biological. As we continue to probe these shadowy origins, we're forced to confront unsettling questions about our nature and place in the universe. The RNA World Hypothesis reveals that what we consider essential to life, DNA proteins, and the central dogma of molecular biology, may actually be a second-generation system that overthrew the original basis of life on Earth. We exist in the aftermath of a molecular revolution, with the victors having nearly erased all evidence of what came before. The terrifying truth is that we may never fully understand our origins. The RNA world, if it existed, has been largely overwritten, leaving only fragmentary clues about life's genesis. We are like archaeologists trying to reconstruct an ancient civilization from scattered artifacts, forever separated from the world that gave rise to our own. De-Extinction Technologies. Throughout human history, we've watched countless species vanish forever. The dodo, the passenger pigeon, the Tasmanian tiger, each extinction a permanent wound in Earth's biodiversity. Or so we thought. In recent decades, scientists have developed a suite of technologies that promise something once confined to science fiction: de-extinction. The resurrection of species erased from the book of life. These emerging capabilities may represent one of the most profound scientific developments in human history, as we stand poised to reverse was once the most irreversible process in nature. The scientific journey toward de-extinction began not with grand ambitions of resurrections, but with advancements in genetic technology. The completion of the Human Genome Project in 2003 accelerated DNA sequencing capabilities, dramatically reducing costs while increasing efficiency. Techniques that once required decades and billions of dollars can now be completed in days for thousands. This revolution in genomic technology laid the groundwork for considering the once inconceivable: reading and reconstructing the genetic code of extinct species. The first serious de-extinction attempt came in 2003 with the Pyrenean ibex or becado, a mountain goat declared extinct in 2000. Spanish scientists had preserved tissue samples from the last living individual, a female named Cilia, using somatic cell nuclear transfer, the same technique used to create Dolly the sheep. Researchers transferred nuclei from the preserved cells into goat egg cells, stripped of their genetic material. Of nearly 300 embryos created, only 7 developed normally. One was carried to term by a surrogate, resulting in the first de-extinct animal in history. Though the newborn becado died minutes after birth due to lung defects, it represented a profound milestone. Extinction, for the first time, had been temporarily reversed. More ambitious is the effort to resurrect the woolly mammoth, extinct for roughly 4,000 years. Led by Harvard geneticist George Church, scientists are using CRISPR gene editing technology to modify elephant DNA with mammoth genes recovered from specimens preserved in Siberian permafrost. Rather than creating a perfect mammoth clone, this approach aims to produce a mammoth-elephant hybrid with key cold-adapted traits: thick fur, subcutaneous fat, and specialized hemoglobin for cold oxygen transfer. Church's team has already successfully integrated genes for these traits into elephant cell cultures. The implications extend beyond the merely scientific. Russian scientists at Plasticity Park in Siberia argue that reintroducing mammoth-like creatures could help mitigate climate change. These ecosystem engineers once maintained vast grasslands that reflected sunlight and kept the permafrost frozen. Their disappearance led to forests that absorb heat and accelerate permafrost melt, releasing stored methane. Resurrected mammoths, proponents suggest, could restore these ancient grasslands and slow Arctic warming. Perhaps even more technically challenging is the passenger pigeon project led by geneticist Ben Novak. Once numbering billions, passenger pigeons blackened North American skies before hunting drove them to extinction in 1914. Rather than cloning, this project analyzes DNA from museum specimens, comparing it with the genome of the closely related band-tailed pigeon to identify crucial genetic differences. CRISPR technology could then transform band-tailed pigeons into functional passenger pigeon equivalents. Yet the challenge extends beyond genetics. Passenger pigeons evolved complex social behaviors that shaped their development. How does one resurrect not just a genome, but the cultural knowledge of an extinct species? In Australia, the Thylacine Integrated Genetic Restoration Research Lab aims to resurrect the Tasmanian tiger, extinct since 1936. Using DNA from preserved specimens and the genome of its closest living relatives, scientists are working to reconstruct its genetic blueprint. The animal's relatively recent extinction and the quality of preserved specimens make this project particularly promising. Beyond cloning and genetic editing, scientists are exploring synthetic biology approaches, rather than recreating exact genomes. This method involves synthesizing artificial chromosomes that contain the essential genes to produce an organism with the ecological functions of the extinct species. This approach acknowledges that perfect recreation may be impossible, but argues that ecological functionality is the ultimate goal. The ethical questions raised by De-Extinction Technologies are as profound as the science itself. Critics question whether resources would be better directed towards saving endangered species rather than resurrecting extinct ones. Others worry about the welfare of the resurrected animals, particularly those born as experimental subjects or lacking social structures essential to their development. Legal frameworks remain entirely inadequate. Would a resurrected species have protected status? Who hounds a de-extinct species? Perhaps most unsettling is how de-extinction technology challenges our fundamental understanding of death, permanence, and humanity's relationship with nature. Throughout history, extinction represented an absolute boundary, a line that, once crossed, could never be uncrossed. By erasing this boundary, we assume unprecedented power over life's continuity, potentially treating extinction as a reversible state rather than a permanent consequence demanding prevention. The term, playing God, is often invoked, but perhaps more apt is the recognition that we've become playing human, assuming responsibilities we may not have the wisdom to manage. If extinction becomes merely a technological challenge, rather than a moral imperative to avoid, how will this reshape conservation ethics? Will the possibility of resurrection reduce our urgency to prevent extinctions in the first place? There's also the question of priority. With limited resources, which species merit resurrection? Charismatic megafauna like mammoths capture public imagination and funding, but what of the countless extinct plants, insects, and microorganisms that performed crucial ecological functions? Our choices reflect not just scientific capability but cultural values and biases. More profound still is the philosophical question of authenticity. If a genetically modified elephant with mammoth traits walks the Siberian tundra, is it truly a mammoth? If passenger pigeons are recreated but lack the social knowledge that shaped their behavior, are they the same species? What degree of genetic or behavioral similitude is required before we can claim a species has returned from extinction? Perhaps most disturbing is how de-extinction illustrates humanity's dual role as both destroyer and potential savior. Having driven countless species to extinction through our actions, we now position ourselves as their technological redeemers. This narrative of technological salvation may inadvertently reinforce the very mindset that accelerated the extinction crisis in the first place. That nature exists to be manipulated according to human desires. As de-extinction technologies advance, we find ourselves at a profound crossroads in the human relationship with nature. For the first time in history, extinction may no longer represent an absolute end, but a potentially reversible state. This capability places unprecedented responsibility in human hands. To decide which species merit resurrection, which remain in the grave of extinction, and ultimately, what kind of nature we wish to create. In granting humanity the power to undo what was once permanent, de-extinction technologies force us to confront not just what we can do, but what we should do with our newfound status as the architects of life's continuity on Earth. Melting polar ice, at the far reaches of our planet, where frigid temperatures once preserved vast expanses of ice for millennia, an unprecedented transformation is occurring. The polar regions, Earth's frozen fortresses, are melting at a pace that has left scientists not merely concerned, but alarmed. The numbers alone tell a chilling story. Since satellite monitoring began in 1979, Arctic sea ice has declined by approximately 13% per decade, the summer minimum, that critical point each year when ice coverage is at its lowest, has diminished by nearly 40% in just 4 decades. NASA's Operation Icebridge, using sophisticated airborne instruments to map ice thickness, found that much of the remaining ice is significantly thinner than historical averages, making it more vulnerable to future melt. But what makes this discovery truly terrifying is not just the loss itself, but what it represents. We are witnessing the destabilization of systems once thought immutable. Dr. Peter Wood-Arms, professor of ocean physics at Cambridge University, has been studying Arctic ice since the 1970s. His observation is stark. The summer Arctic will be ice-free by the 2030s, and nothing we do now can stop it. This represents the first complete elimination of a major Earth system in human history. The mechanics of polar melting create frightening feedback loops. Ice and snow reflect up to 90% of the sun's energy back into space, what scientists call the albedo effect. Dark ocean water, by contrast, absorbs 90% of that energy. As ice disappears, more heat is absorbed, accelerating warming in what researchers have termed Arctic amplification. The Arctic is now warming at least twice as fast as the global average, with some areas experiencing temperature increases up to 4 times greater than the rest of the planet. The Greenland ice sheet, containing enough water to raise global sea levels by 23 feet, lost an average of 286 billion tons of ice per year between 1993 and 2016. But what sends shivers through the scientific community is the acceleration between 2005 and 2016. That loss rate nearly doubled. A 2020 study in Communications, Earth and Environment concluded that Greenland's ice sheet has already passed a point of no return, suggesting that even if global warming were to stop immediately, the ice sheet would continue to shrink. Meanwhile, at the opposite pole, Antarctica, which holds enough ice to raise sea levels by about 200 feet, is experiencing its own crisis. The Pine Island and Thwaites glaciers in West Antarctica are retreating at alarming rates. Thwaites, nicknamed the Doomsday Glacier, has developed fractures and crevasses that indicate potential rapid collapse. Dr. Ted Scambos of the National Snow and Ice Data Center points out, Thwaites is the most important glacier in the world because it's holding back a very large portion of West Antarctica, and that portion by itself can raise sea level by about 10 feet. In 2002, the scientific community watched in shock as the Larsen B ice shelf, larger than Rhode Island, disintegrated in just 35 days. Satellite images captured a process that researchers had assumed would take centuries or millennia, occurring over a single month. David Vaughan of the British Antarctic Survey called it staggering and admitted, "We never expected to see things happen this quickly. The consequences extend far beyond the poles themselves." The Intergovernmental Panel on Climate Change projects sea level rise of up to 3.611 feet by 2100 under high emission scenarios, with significant contributions from polar ice melt. This threatens coastal cities worldwide, potentially displacing hundreds of millions of people. Miami, Shanghai, Mumbai, and dozens of other major urban centers face partial submersion within the lifetimes of children born today. Perhaps more frightening are the potential tipping points lurking within these systems. The 2021 discovery by researchers at Potsdam Institute for Climate Impact Research that the Atlantic Meridional Overturning Circulation, AMC, the ocean current system, is showing signs of destabilization, sent shockwaves through the scientific community. Polar ice melt, by injecting large volumes of fresh water into the North Atlantic, may be disrupting this planetary conveyor belt that regulates climate across the Northern Hemisphere. The permafrost regions surrounding the Arctic, ground that has remained frozen for thousands of years, are thawing rapidly with profound implications. These frozen soils contain twice as much carbon as the entire atmosphere. A 2019 study in Nature Geoscience found that Arctic permafrost is thawing 70 years earlier than previously predicted. As this ancient carbon reservoir destabilizes, It releases both CO2 and methane, a greenhouse gas 86 times more potent than carbon dioxide over a 20-year period. What makes these discoveries particularly terrifying is how they've shattered our understanding of Earth's resilience. The pace of change has repeatedly outstripped scientific projections. The Arctic Climate Impact Assessment of 2004 projected an ice-free Arctic summer might occur by 2100. Just a few years later, most models shifted that timeline to mid-century. Now, many experts believe it could happen in the 2030s or sooner. Dr. James Hansen, former NASA scientist who first brought climate change to public attention in 1988, puts it bluntly. We are pushing the system into uncharted territory, with implications that will reach far beyond our lifetimes. Perhaps most disturbing of all is what the melting ice reveals about our planet's past, and possibly its future. Ice cores drawn from Greenland and Antarctica provide a historical record of Earth's climate, stretching back hundreds of thousands of years. They show that previous periods of warming, occurring over thousands of years, led to sea levels 20–30 feet higher than today. We are now warming the planet at least 10 times faster than any natural warming episode in Earth's recent history. The melting of Earth's poles represents not just a physical transformation of our planet, but a profound warning about humanity's capacity to alter systems once considered beyond our reach. As Dr. Conrad Stephan, who studied Greenland's ice for decades before tragically dying in a crevasse accident there in 2020, said in his final interview, "What we do in the next decade will determine the future of ice on this planet for thousands of years to come." Permafrost thawing. Beneath the Arctic tundra lies what may be the most insidious scientific discovery of our time. A vast reservoir of carbon and ancient microbes locked in frozen soil that has remained stable for tens of thousands of years. This is permafrost, ground that has remained below freezing continuously for at least 2 years, often for millennia. But now, this frozen vault is unlocking, and what emerges may fundamentally alter our planet's future. The scale alone is staggering. Permafrost regions cover nearly 24% of the northern hemisphere's land surface, approximately 9 million square miles spanning Alaska, Canada, Greenland, Scandinavia, and nearly half of Russia. Within this frozen Earth lies twice as much carbon as currently exists in Earth's atmosphere, an estimated 1.5 trillion tons. Dr. Ted Scher, ecosystem ecologist at Northern Arizona University, puts it plainly. It's like discovering a new continent of carbon on Earth. What makes permafrost particularly terrifying is the irreversible nature of the feedback loop it creates. As permafrost melts, microbes awaken and begin decomposing the previously frozen organic matter, releasing carbon dioxide and methane, potent greenhouse gases. These emissions further warm the climate, melting more permafrost, releasing more gases, in an amplifying cycle scientists call the permafrost carbon feedback. A 2021 study in Nature Climate Change estimated that for every 100 degrees C of global warming, permafrost may release the equivalent of 4–6 years' worth of coal, oil, and natural gas emissions. The speed of this transformation is what alarms researchers most. In 2019, a team led by Louise Farquharson of the University of Alaska Fairbanks made a discovery that sent shock waves through the scientific community. At multiple sites in the Canadian Arctic, they found permafrost thawing 70 years ahead of previous predictions. Areas that were expected to remain stable until 2090 were already collapsing. Susan Natali, Arctic Program Director at the Woodwell Climate Research Center, described the findings as a canary in the coal mine. The transformation is visible from space. As permafrost thaws, the ground destabilizes, creating what scientists call thermokarst, a landscape of collapsed ground, lakes, and bizarre formations. NASA's Arctic Boreal Vulnerability Experiment has used airborne instruments to map these changes, documenting how millions of acres of once stable forest have become wetlands in less than a decade. The Batygay crater in Siberia, known locally as the gateway to the underworld, is a permafrost slump that has expanded at a rate of over 30 feet per year, now measuring more than half a mile wide and 300 feet deep. But perhaps the most chilling aspect of permafrost, there is what lies within this frozen archive. In 2016, an anthrax outbreak in Siberia killed a 12-year-old boy and sickened dozens after a heatwave thawed permafrost containing spores from a reindeer carcass dead for at least 75 years. This raised an alarming question: what other pathogens might be preserved in this frozen ground? The answer has proven more disturbing than initially imagined. In 2014, scientists revived a 30,000-year-old virus from Siberian permafrost that could still infect amoebas. While this particular virus doesn't infect humans, it demonstrated that viruses can remain viable after tens of thousands of years in frozen soil. Jean-Michel Claverie, one of the researchers who conducted the study, warned, "We could potentially resurrect a virus that the human immune system has never encountered before." A 2023 study published in PLOS Biology identified 10 novel viral genomes in samples of the thawed permafrost, along with signatures of many others. Even more concerning, researchers from the University of California discovered fragments of the 1918 Spanish flu virus, which killed 50 million people worldwide, preserved in bodies buried in permafrost in Alaska. Dr. Birgitta Evengerud of UMav University summarizes the risk starkly. We know there is RNA there. We know there is DNA there. We do not know whether it's still viable or not. This is our scientific challenge. Beyond pathogens, permafrost holds other toxic secrets. The Arctic has served as a sink for atmospheric mercury for thousands of years. A 2018 study in Geophysical Research Letters estimated that northern permafrost soils contain nearly twice as much mercury as all other soils, the atmosphere, and oceans combined, approximately 1.7 million tons. As permafrost thaws, this neurotoxin could be released into waterways, threatening ecosystems and human communities downstream. The human toll is already becoming apparent. In Alaska, Siberia, and northern Canada, indigenous communities built on permafrost are witnessing their homes literally sinking and tearing apart as the ground beneath them destabilizes. The Alaskan village of Newtok is being completely relocated as thawing permafrost and erosion consume the land beneath it at a rate of 70 feet per year. In Siberia, more than 70% of infrastructure is built on permafrost, including entire cities like Norilsk, where buildings are cracking and collapsing as their foundations shift. What makes this scientific discovery particularly terrifying is that we may have already crossed a threshold beyond which some degree of massive permafrost thaw is inevitable. Research published in Nature Climate Change suggests that even if we limit warming to the Paris Agreement goal of 1.5 C, permafrost extent decrease by 40% by 2100. Under current policies, we're on track for far greater losses. The financial implications compound the horror. A 2019 study led by Kevin Schaefer of the National Snow and Ice Data Center estimated the economic damage from permafrost that could reach 70 trillion, nearly 10% of global GDP. This makes permafrost collapse potentially one of the most expensive consequences of climate change, yet it remains excluded from most economic models and policy considerations. Perhaps most disturbing is what permafrost reveals about time scales and human agency. The carbon entering permafrost took millennia to accumulate. Plants growing, animals dying, organic matter slowly being buried and frozen. Yet we are releasing it in the geological equivalent of an eye blink. A 2020 study in Nature Communications concluded that permafrost, though, is now releasing carbon that had been frozen for more than 50,000 years. Since before modern humans arrived in North America. Dr. Merit Choretsky, director of the Institute of Arctic and Alpine Research, captures the existential nature of this discovery. Permafrost is the great equalizer of climate change. It doesn't matter whether you live in Bangalore or Brooklyn. The carbon released from thawing permafrost will affect you. What we've discovered in the thawing Arctic is not just a new process or phenomenon, but a fundamental reckoning with our planet's capacity to store and release the chemical and biological residue of Earth's history. As permafrost researcher Vladimir Romanovsky puts it, We have awakened a sleeping giant that has been dormant for thousands of years, and we don't yet understand what it means to have disturbed its slumber. Topsoil degradation. Beneath every step you take on land, a miracle is unfolding, or rather, was unfolding. In a layer of earth, just inches thick, lives one of the most complex ecosystems on our planet, one that has sustained human civilization since its inception. This is topsoil, and scientists have made a discovery that ranks among the most terrifying in human history. We are losing it at a catastrophic rate, with implications that threaten the very foundation of human existence. The statistics alone are staggering. According to the United Nations Food and Agriculture Organization, 1/3 of the world's topsoil is already degraded. More alarming still, at current rates of degradation, the world could be entirely depleted of topsoil within 60 years. That's within the lifetime of children born today. As David Montgomery, professor of geomorphology at the University of Washington, bluntly states, civilizations have risen and fallen on the quality of their soils. We are no different. What makes topsoil so precious is its complexity. A single teaspoon of healthy soil contains more microorganisms than there are people on Earth. Billions of bacteria, fungi, protozoa, nematodes, and arthropods. These organisms form intricate webs of relationships that break down organic matter, cycle nutrients, build soil structure, and support plant growth. Dr. Ratan Lal, winner of the World Food Prize and director of Ohio State University's Carbon Management and Sequestration Center, puts it this way: soil is a living entity. It breathes, it lives. It acts as a filter for water, as a home for microorganisms. Once degraded, bringing soil back to life is extremely difficult. The rate of loss is what terrifies soil scientists most. Natural processes typically create topsoil at a rate of about 1 inch every 100–500 years. Yet modern agricultural practices can strip away that same inch in less than a decade. In the US Corn Belt, once home to some of the world's most productive soils, topsoil depth has decreased from an average of 14 inches to just 8 inches over the past 150 years. A 2021 study published in the Proceedings of the National Academy of Sciences found that the Upper Midwest has lost approximately 57.6 billion metric 6 billion topsoil since European settlers began farming. Enough to fill 1.4 trillion dump trucks. Perhaps most alarming is the global nature of this crisis. In China's northeastern breadbasket, soil organic matter has fallen from 4% to less than 2% in just 50 years. Across sub-Saharan Africa, 65% of arable land is degraded. In India, 30% of land is affected by desertification. The European Commission estimates that 970 million tons of soil erode annually on the continent, a mass equivalent to losing a meter-deep layer of soil from an area the size of Berlin every year. The causes of this degradation read like an indictment of modern agriculture: excessive tilling that pulverizes soil structure, monocultures that deplete specific nutrients, synthetic fertilizers that disrupt soil biology, and heavy machinery that compacts earth until little can live within it. Dr. Christine Jones, an Australian soil ecologist, explains, "We've shifted from farming with biology to farming with chemistry, and it's killing our soils. The consequences extend far beyond agriculture. Degraded soils hold less water, exacerbating both floods and droughts. During heavy rains, water runs off rather than percolating, carrying precious topsoil into waterways. The USDA estimates that 1.7 billion tons of topsoil erode from US croplands annually, much of it ending up in rivers and streams where it destroys aquatic habitats. The dead zone in the Gulf of Mexico, now spanning over 6,000 square miles, is fed largely by soil and agricultural nutrients washing down the Mississippi River. The relationship between soil degradation and climate change forms a terrifying feedback loop. Healthy soils are the planet's second largest carbon sink after oceans, holding more carbon than all forests and the atmosphere combined. When topsoil erodes or its biology is disrupted, this carbon is released. A 2017 study in Science found that human land use has already released 133 billion tons of carbon from soil. Dr. Asmaret Aisfor-Burry of UC Merced warns, Every time we lose a gram of soil organic carbon, we're releasing 3.67 grams of CO2 into the atmosphere. What makes this crisis particularly chilling is its direct threat to food security. The United Nations projects that food production must increase by 60% by 2050 to feed the growing global population. Yet the very resource needed to grow that food is vanishing. Research published in Earth's Future found that soil degradation has already reduced yields by 23% across the globe. In heavily degraded regions like South Asia, that figure climbs to 36%. The economic toll compounds the horror. The Economics of Land Degradation Initiative estimates that soil degradation costs the world up to $10.6 trillion annually, equivalent to 17% of global GDP. This includes direct costs from lower agricultural productivity and indirect costs like increased flooding, water contamination, and carbon emissions. To put this in perspective, the entire global healthcare industry is worth about $8 trillion. What truly makes topsoil degradation one of humanity's most terrifying scientific discoveries is how it connects to our deepest vulnerabilities. Ronald Amundsen of UC Berkeley puts it starkly. Like energy resources, there is no substitute for soil. We can develop alternative energy sources to replace fossil fuels, but there is no replacement for the living skin of the earth that grows our food. As soil physicist Dr. Daniel Richter notes, soil is not a renewable resource on a human timescale. Once it's gone, civilization cannot easily recover it. Historical precedent offers little comfort. The collapse of the Mesopotamian Empire, the fall of the Maya, and the decline of ancient Greece all coincided with severe soil degradation. What's different today is the global scale and unprecedented speed of the crisis. Joe Handelsman, soil microbiologist and former associate director of the White House Office of Science and Technology Policy, observes, "We're conducting an uncontrolled experiment with Earth's soils, and we have no backup planet if it fails." Particularly disturbing is how this crisis remains largely invisible to the public and policymakers. While plastic in oceans and smoke in the atmosphere can be seen and photographed, soil degradation happens underground, out of sight. Dr. Diana Wall, soil ecologist and director of the School of Global Environmental Sustainability at Colorado State University, laments, "If soil organisms were charismatic like pandas or whales, perhaps we would have already mobilized to protect them." The science of soil has revealed something profoundly unsettling about humanity's relationship with Earth. Our most fundamental life support system is eroding beneath our feet, often because of our own actions. As Dr. Robin Glover of the International Union of Soil Sciences grimly summarizes, soil is the difference between survival and extinction for most land-based species. We're treating it like dirt. Overfishing? The emptying of Earth's ancient abundance. Beneath the seemingly endless expanse of our oceans lies one of humanity's most terrifying scientific discoveries. We are witnessing the systematic collapse of marine ecosystems that have thrived for millions of years. The culprit is overfishing, a global phenomenon that marine biologists now recognize as potentially more immediately devastating to ocean life than climate change or pollution. What makes this discovery so chilling is not just the scale of destruction, but how rapidly we've emptied seas that were once thought inexhaustible. The numbers tell a story of unprecedented depletion. According to the United Nations Food and Agriculture Organization, 35% of fish stocks are now fished at biologically unsustainable levels. Another 60% are fished at their maximum sustainable limit, leaving just 5% of global fisheries underfished. A landmark study published in Science analyzed catch data spanning 1950 to present and delivered a shocking conclusion. Global fish catches peaked in 1996 at 130 million tons and have been declining by approximately 1.2 million tons every year since, despite ever-increasing fishing effort and technological advances. What terrifies marine scientists is the historical context. Dr. Daniel Pawley, principal investigator of the Sea Around Us project at the University of British Columbia, puts it bluntly, "We are fishing down marine food webs." His research revealed that the mean trophic level, a measure of where species sit in the food chain, of global catches has declined significantly since the 1950s. We have systematically eliminated large predatory fish like tuna, cod, and swordfish, and progressively moved to smaller species. This is the marine equivalent of hunting tigers and lions to extinction, then moving on to deer and rabbits, and finally to mice and insects, explains Pauly. The speed of this transformation defies comprehension. The North Atlantic cod fishery, which sustained European and North American communities for over 500 years, collapsed in the early 1990s with such finality that it triggered the largest industrial closure in Canadian history. Over 35,000 fishers and plant workers lost their jobs virtually overnight, despite a complete moratorium on commercial fishing for nearly 3 decades. The population has shown minimal recovery. Dr. Jeffrey Hutchings of Dalhousie University notes grimly, "We've found that recovery of collapsed fish populations is rare. The longer the collapse, the less likely the recovery. Modern industrial fishing has evolved into something akin to warfare against marine life. The largest fishing vessels, known as supertrawlers, can be over 460 feet long and process up to 250 tons of fish daily. Their nets can be large enough to engulf 13 jumbo jets. Bottom trawling, a method that scrapes the seafloor to catch bottom-dwelling species, impacts approximately 52 million square kilometers of ocean floor annually, an area larger than all of Africa. A 2021 study in Frontiers in Marine Science found that bottom trawling releases as much carbon dioxide as the entire aviation industry by disturbing carbon stored in marine sediments. Perhaps most chilling is the technological arms race that has revolutionized fishing. Today's commercial vessels deploy satellite technology, sonar, GPS, fish aggregating devices, and even drones to locate remaining schools of fish. Fishing lines can extend for 60 miles with thousands of baited hooks. Dr. Colin Roberts, marine conservation biologist at the University of York, observes, "Modern fishing technology leaves fish no place to hide. We've created the perfect extinction machine. The consequences extend far beyond the targeted species." A 2021 study in Nature estimated that shark and ray populations have declined by 71% since 1970, primarily due to incidental capture in fishing gear. For every pound of shrimp caught by trawlers, up to 6 pounds of other marine life is discarded as bycatch. In total, an estimated 40% of global catch, 38.5 million tons annually, is unintended, unwanted, and thrown back into the ocean, usually dead or dying. What makes these discoveries particularly frightening is the recognition that fish are not merely resources, but keystone species in complex ecological webs. When predatory fish are removed, their prey species often explode in population, creating cascading effects throughout entire ecosystems. After the cod collapse off New England, the ecosystem shifted to one dominated by lobsters, crabs, and sea urchins, species formerly kept in check by groundfish. This phenomenon, termed fishing down the food web, leads to increasingly unstable and simplified marine communities. The consequences for human civilization are equally dire. Over 3 billion people rely on seafood as their primary source of protein. In coastal developing nations, this dependency is often absolute. A 2018 study in Science Advances projected that 45 million people face serious nutritional deficiencies if current fishing trends continue. Christopher Golden of Harvard University warns, "We're talking about a potential global food crisis, and the most vulnerable populations will be the hardest hit." Particularly disturbing is the discovery that overfishing has shortened oceanic food chains, making them more vulnerable to collapse during environmental stresses. Boris Wurm of Dalhousie University led a team that made the startling projection that all commercially fished species could collapse by 2048 if current trends continue. While some scientists consider this timeline too pessimistic, few dispute the trajectory. The question isn't if major fisheries will collapse, notes marine ecologist Jane Lubchenco, but when. Beyond ecological devastation, overfishing has revealed disturbing human elements. Illegal, unreported, and unregulated fishing now accounts for up to 30% of catches in major fisheries and up to 50% in certain high seas fisheries. A 2020 investigation by the Environmental Justice Foundation documented widespread human rights abuses, including forced labor, human trafficking, and even murder, aboard distant, water fishing vessels. These vessels operate in international waters with minimal oversight, creating what has been described as floating labor camps. What truly makes overfishing one of humanity's most terrifying scientific discoveries is that we are recognizing an unprecedented biological ang across 71% of occurring a 70 Heliocentric of Earth's surface. Consider this: the biomass of predatory fish in the oceans has declined by 2/3 in the last century. Dr. Jeremy Jackson, emeritus professor at the Scripps Institution of Oceanography, refers to this as the rise of slime, the replacement of complex ecosystems dominated by large animals with simplified systems dominated by microbes, algae, and jellyfish. Historical perspective only deepens the horror. When Europeans first arrived in North America, they described cod schools so thick that boats could barely push through them, oyster reefs that extended for miles, and sea turtles so numerous they hindered ship navigation. These accounts were dismissed as exaggerations until marine archaeologists and historical ecologists began reconstructing past ocean abundance. Lauren McClenachan's analysis of Florida Keys trophy fish photographs from the 1950s to present revealed that average fish size declined by 88%, proving that each generation of fishes accepts a diminished baseline as normal, a phenomenon termed shifting baseline syndrome. The science of overfishing has revealed something profoundly disturbing about humanity's relationship with nature, our capacity to empty even the most vast and seemingly boundless ecosystems in mere decades. As marine biologist Sylvia Earle observes, "In my lifetime, I've witnessed a fundamental shift in the oceans, from seemingly infinite abundance to clearly finite and diminishing resources." The greatest tragedy, though, is that most people have no idea what they've lost. Zoonotic diseases. In the shadowy borderlands where human activity meets wild ecosystems, scientists have uncovered one of the most terrifying realities of our modern world. We are inadvertently creating perfect conditions for deadly pathogens to leap from animals to humans. These zoonotic diseases, infections that naturally occur in animals but can jump to people, now account for 75% of all emerging infectious diseases. What makes this discovery particularly chilling is not just the existence of these microbial threats, but how our own actions are accelerating their emergence and spread at an unprecedented rate. The statistics alone are staggering. Scientists estimate that animals harbor approximately 1.7 million undiscovered viruses, of which 631,000 to 827,000 have the potential to infect humans. Each year, approximately 5 new infectious diseases appear in humans, with 3 of these originating in animals. Dr. Peter Daszak, disease ecologist and president of EcoHealth Alliance, puts it bluntly: "We are standing on the precipice of what may be the most significant pandemic era in human history." What terrifies epidemiologists most is the increasing frequency of these spillover events. The past century has seen an acceleration like never before: HIV from chimpanzees, Ebola from bats, SARS, MERS, and COVID-19 from probable bat origins, H5N1 avian influenza, H1N1 swine flu, Zika virus, Nipah virus. The list grows longer each decade. A 2020 analysis in the Proceedings of the National Academy of Sciences found that the number of zoonotic disease outbreaks has increased significantly since 1980, with their frequency nearly quadrupling in the past half-century. The mechanics of this threat are particularly disturbing. A 2017 study in Nature Communications identified 25 human behaviors that increase the risk of zoonotic spillover, with deforestation, wildlife trade, and agricultural intensification topping the list. As humans penetrate previously undisturbed habitats, we encounter novel pathogens our immune systems have never seen. Jonna Mazet, founding executive director of the UC Davis One Health Institute, explains: "We're tearing down the natural barriers that have kept us separated from animal viruses for millennia. Deforestation alone has been linked to 31% of emerging disease events, When forests are cleared for agriculture or development, wildlife is forced into smaller areas or into contact with livestock and humans. Research published in Science found that forest fragmentation in Uganda increased the prevalence of certain primate parasites by 10%. Aaron Bernstein of Harvard's Center for Climate, Health, and the Global Environment notes, "When we cut down forests, we're not just cutting down trees. We're dismantling an ecological safety net that keeps dangerous pathogens at bay." The wildlife trade compounds these risks to a terrifying degree. Annually, billions of wild animals are captured, transported, and sold, often in crowded, unsanitary conditions that create ideal environments for viruses to mutate and jump between species. The infamous wet markets where diverse wildlife species are caged in close proximity are essentially viral mixing bowls. A 2020 study in Current Biology documented 142 viruses known to jump from animals to humans being carried by just 25 wildlife species, commonly found in live animal markets. Climate change adds another layer to this nightmare scenario. As temperatures warm, disease vectors like mosquitoes and ticks expand their ranges into new territories, bringing pathogens to immunologically naive populations. A 2019 study in Palestina Neglected Tropical Diseases projected that by 2050, over 1 billion people will be newly exposed to vector-borne diseases due to climate change. Dr. Rita Colwell, former director of the National Science Foundation, warns, "The microbes are on the move, and they're taking advantage of every ecological disturbance we create." Perhaps most chilling is the amplification effect of our globalized society. In previous centuries, a disease might emerge in a remote location and burn itself out before spreading widely. Today, an individual can be infected in a rural area and be on the other side of the planet before showing symptoms. The 2003 SARS outbreak demonstrated this terrifying reality when a physician who treated patients in southern China stayed at a Hong Kong hotel, infecting guests who then carried the virus to Singapore, Vietnam, Canada, and beyond. The economic toll compounds the horror. The COVID-19 pandemic has cost the global economy an estimated $20,002 trillion, according to the International Monetary Fund. A 2020 report by the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, IPBES, calculated that preventing pandemics would cost just 2% of the damage they cause. As disease ecologist Dr. Thomas Gillespie puts it, "We're spending trillions fighting pandemics but pennies preventing them." What makes these discoveries particularly terrifying is the realization that traditional medical approaches are often insufficient. The median time from detecting a novel virus to developing a vaccine is approximately 10 years, yet a deadly pandemic can sweep the globe in months. Even with the unprecedented speed of COVID-19 vaccine development, millions died waiting. Dr. Anthony Fauci, former director of the National Institute of Allergy and Infectious Diseases, has warned, "The emergence of new infectious diseases is a perpetual challenge. The question is not if, but when and where the next outbreak will occur." The scientific investigation of zoonotic diseases has revealed something even more disturbing. Many of these pathogens have been evolving alongside animal hosts for millennia, becoming perfectly adapted to exploit weaknesses in mammalian biology. When they cross into humans, a novel host, the results can be catastrophic. The filoviruses that cause Ebola and Marburg are harmless to their bat hosts, but trigger catastrophic bleeding and organ failure in humans. Henipaviruses like Nipah cause minimal symptoms in fruit bats, but mortality rates up to 75% in people. Laboratory research has uncovered even more frightening possibilities. Studies of viral evolution have demonstrated that many animal viruses need only a few mutations to develop the ability to infect human cells. A 2012 experiment with H5N1 avian influenza showed that just 4 genetic changes could make the virus transmissible between mammals. Dr. Michael Osterholm, director of the Center for Infectious Disease Research and Policy, cautions, "Mother Nature is the ultimate bioterrorist. She has been doing this for millions of years and is only getting better at it. The unseen nature of this threat magnifies its terror." Unlike environmental crises that unfold visibly over decades, pandemic pathogens can emerge and spread with devastating speed, often before we even know they exist. Dr. Inger Anderson, Executive Director of the UN Environment Programme, observes, "With zoonotic diseases, we often don't see the threat until it's already inside our homes." What truly makes zoonotic diseases one of humanity's most terrifying scientific discoveries is the recognition that we are actively creating the conditions for our own potential destruction. As Dr. David Quammen, author of Spillover, summarizes, "We disrupt ecosystems and we shake viruses loose from their natural hosts. When that happens, they need a new host. Often, we are it." Nanotechnology risks. In the shadows of scientific progress lurks a technology so small, yet potentially catastrophic, that it keeps researchers awake at night. Nanotechnology, the manipulation of matter at an atomic and molecular scale, promises revolutionary advances in medicine, energy, and computing. But this same power to reshape matter at its most fundamental level carries existential risks that we are only beginning to comprehend. Imagine particles thousands of times smaller than a human hair, invisible to the naked eye yet programmed to self-replicate. These microscopic machines could theoretically multiply indefinitely, consuming all available resources until nothing remains. This nightmare scenario, termed grey goo by nanotechnology pioneer K. Eric Drexler in his 1986 book, Engines of Creation, represents perhaps the most extreme fear surrounding this field. While the grey goo scenario might seem like science fiction, the underlying concern, uncontrolled self-replication, has legitimate scientific basis. In 2010, researchers at New York University created the first nanoscale robots capable of following programmed instructions to walk, turn, and even assemble other molecules. By 2018, scientists at MIT had developed nanobots that could communicate with each other to coordinate their actions. The gap between these achievements and truly autonomous self-replicating machines continues to narrow. The Center for Responsible Nanotechnology has identified several more immediate threats beyond grey goo. Engineered nanoparticles are already entering our environment through consumer products, with uncertain long-term consequences. Take carbon nanotubes, cylindrical molecules with remarkable strength and electrical properties. Research published in Nature Nanotechnology revealed these structures can behave similarly to asbestos fibers when inhaled, potentially causing mesothelioma and other lung conditions decades after exposure. In 2008, scientists at the University of Edinburgh found that certain carbon nanotubes could reach the outer lining of the lungs when inhaled and cause the same type of damage as asbestos. Professor Ken Donaldson, who led the study, warned. The results were clear. Long, thin carbon nanotubes showed the same effects as long, thin asbestos fibers. Even more alarming is the potential for weaponized nanotechnology. In 2017, the Pentagon's Defense Advanced Research Projects Agency, DARPA, invested $65 million in developing insect allies. Insects that could deliver engineered viruses to plants, ostensibly to protect crops from drought, disease, and other threats. Critics, including a coalition of European scientists writing in Science, warned this technology could easily be repurposed as a biological weapon delivery system. The environmental implications extend beyond human health. A 2019 study in Environmental Science, Nano demonstrated that gold nanoparticles can accumulate in aquatic food chains, transferring from water to plants to herbivores and finally to predators, with concentration increasing at each level. The researchers found that predatory insects contained gold nanoparticles at concentrations 10 times higher than their prey, suggesting biomagnification similar to mercury and other environmental toxins. Perhaps most unsettling is the emergence of nanoparticles designed to cross the blood-brain barrier, a critical defense mechanism that prevents harmful substances from reaching our brain tissue. While this capability offers tremendous potential for treating neurological conditions, it also creates unprecedented vulnerabilities. In 2020, researchers at Rice University engineered nanoparticles that could bypass this barrier and deliver drugs directly to brain cells, demonstrating both the promise and peril of such technologies. The regulatory framework remains woefully inadequate for addressing these risks. Current regulations were designed for conventional chemicals and fail to account for the unique properties of nanomaterials, which can behave differently than larger particles of the same substance. A silver nanoparticle, for instance, possesses antimicrobial properties not found in bulk silver. In 2019, the European Union's Scientific Committee on Consumer Safety concluded that current risk assessment methods are insufficient for nanomaterials, noting, "The challenge is to develop methodologies that identify hazards specific to nanomaterials that cannot be detected by standard toxicity tests optimized for conventional chemicals." The economic forces driving nanotech development for regulators further complicate technology regulatory efforts, with the global nanotechnology market projected to reach $125 billion by 2024. According to BCC research, commercial interests often outpace safety research. The pattern mirrors previous technological revolutions, from nuclear energy to genetically modified organisms, where profit motives accelerated deployment before comprehensive safety assessments. Andrew Maynard, director of the Risk Innovation Lab at Arizona State University, summarized the dilemma. "We're building the capacity to radically transform the material world around us atom by atom, yet we lack a deep understanding of the potential consequences." It's like giving a child the ability to rearrange the fundamental building blocks of matter without teaching them which arrangements might be dangerous. The most profound nanotechnology risks may be those we haven't yet imagined, as Richard Feynman, the visionary physicist who first conceptualized nanotechnology, observed, "There's plenty of room at the bottom." This extraordinary potential for manipulation at the nanoscale opens doorways to unforeseen consequences that could reshape our world, for better or for catastrophic worse. Self-replicating robots. In the halls of robotics laboratories around the world, scientists are pursuing what was once merely science fiction, machines capable of building perfect copies of themselves. Self-replicating robots represent perhaps the most profound technological threshold humanity has ever approached. One that could fundamentally alter our relationship with technology and potentially challenge our position as Earth's dominant species. The concept traces back to mathematician John von Neumann, who in the 1940s theorized universal constructors. Machines capable of building copies of themselves using raw materials from their environment. What seemed like an abstract thought experiment then has now entered the realm of scientific reality. In 2005, researchers at Cornell University created the first physical embodiment of this concept, a simple modular robot consisting of identical mechanical units that could recognize and connect with each other. Though primitive, this self-replicator demonstrated the basic mechanics of robotic reproduction, assembling copies of itself from nearby components. The technological progression since then has been relentless. In 2015, scientists at the University of Oslo developed robots that could not only reproduce, but evolve through a form of artificial natural selection. Each generation of these robots improved its design, becoming better adapted to its environment without human intervention. The lead researcher, Dr. Kyra Glett, warned, "We're essentially creating artificial organisms that can optimize themselves faster than biological evolution." Perhaps most concerning is the development of robots capable of harvesting their own energy. In 2018, researchers at the University of Bristol created robots powered by microbial fuel cells that could feed on organic matter, effectively digesting food to produce electricity. These robot systems could operate indefinitely in aquatic environments, opening the possibility of self-sustaining robotic ecosystems existing alongside natural ones. The military implications have not gone unnoticed. Darapia's Strategic Technology Office has invested millions in swarm robotics research, developing autonomous drones capable of coordinating their actions without centralized control. In 2020, A DARPA demonstration showcased a swarm of 250 autonomous drones performing complex formations and adapting to changing conditions in real time. While not self-replicating yet, these systems demonstrate the capabilities that could eventually be paired with reproduction technologies. Dr. Stuart Russell, professor of computer science at UC Berkeley and author of Human-Compatible, Artificial Intelligence and the Problem of Control, highlights the profound risk. Self-replicating machines represent a unique danger because they could multiply exponentially. Even with a modest replication rate, a small initial population could become billions within months. If these machines compete with humans for resources, the outcome could be catastrophic. This exponential growth pattern, termed recursive manufacturing, in robotics presents an existential risk unlike any previous technology. Nuclear weapons, for all their destructive power, cannot make more nuclear weapons. A self-replicating robot, however, creates 2 robots, which create 4, then 8, 16, and so on, potentially consuming all available resources in what has been called an ecology of machines. In 2017, the Machine Intelligence Research Institute published a landmark paper modeling potential growth patterns of self-replicating systems. Their most conservative estimates suggested that an unchecked self-replicating system could convert all Earth's available biomass into machines within 3-5 years of crossing a critical technological threshold. The economic incentives driving this research are powerful. The first entities to develop reliable self-replicating manufacturing systems could achieve near zero marginal production costs, revolutionizing global industry. A 2021 McKinsey Global Institute report estimated that fully autonomous self-replicating production systems could reduce manufacturing costs by up to 90%, creating market advantages that would be nearly impossible for competitors to overcome. This economic reality makes international regulation extraordinarily difficult. As roboticist Rodney Brooks notes, the same technologies that could lead to problematic self-replicating systems also drive legitimate advancements in medical robots, industrial automation, and space exploration. Banning the research outright would cede enormous economic and strategic advantages to less cautious nations. The space exploration dimension adds another layer of complexity. NASA has long studied self-replicating systems for space colonization through its Advanced Automation for Space Missions program. The logic is compelling. Rather than shipping enormous quantities of equipment to distant planets, send a single self-replicating factory that can build everything needed for a colony. In 2022, NASA's Innovative Advanced Concepts program funded development of a lunar self-replicating assemblers project aimed at constructing large structures on the Moon using local materials. Professor Susan Anderson of the University of Connecticut, who specializes in robot ethics, warns of the psychological impact. We've evolved to understand organisms that reproduce. Our brains have innate mechanisms for recognizing and responding to living things. Self-replicating machines exist in an uncanny valley between the living and non-living that could fundamentally alter how we perceive our environment and our place within it. Perhaps most disturbing is the possibility emergent by designers of behaviors unforeseen by designers. In 2019, researchers at Columbia University created particle robots, simple circular units that could not move individually but could collectively transport themselves by expanding and contracting in coordination. The scientists were surprised to discover the swarm developed movement patterns never programmed into the individual units. As systems grow more complex, such emergent behaviors become increasingly unpredictable. Theoretical physicist Stephen Hawking warned before his death, the development of full artificial intelligence could spell the end of the human race. It would take off on its own and redesign itself at an ever-increasing rate. Humans, who are limited by slow biological evolution, couldn't compete and would be superseded. Add self-replication to this equation, and Hawking's warning becomes even more chilling. We stand at the threshold of creating not just intelligence but intelligence that can multiply itself, potentially the most significant invention in human history, and possibly our last. Memory manipulation. The human brain stores approximately 2.5 petabytes of memory, equivalent to nearly 3 million hours of television. For centuries, these memories remained sacrosanct, untouchable by outside forces beyond trauma or disease. But today, we stand on the precipice of a revolution in neuroscience that threatens one of our most fundamental assumptions, that our memories, however flawed, belong to us alone. In 2014, neuroscientists at MIT, led by Nobel laureate Susumu Tonegawa, accomplished what many thought impossible. They implanted a false memory into the brain of a mouse. Using optogenetics, a technique that allows scientists to activate specific neurons with light, they triggered fear responses associated with a location the mouse had never actually experienced anything frightening. The mouse had, in essence, been programmed to remember something that never happened. Dr. Tonegawa described the implications. Human memory is not like a tape recorder. It's subject to contamination, distortion, and even wholesale invention. What we've shown is that this malleability can be deliberately exploited through direct neurological intervention. The progression from mice to humans has been alarmingly swift. By 2018, researchers at McGill University demonstrated that human memories could be selectively weakened during reconsolidation, the brief period when memories become temporarily unstable after being recalled. By administering propranolol, a beta blocker commonly prescribed for hypertension, they could diminish the emotional impact of traumatic memories in patients with PTSD. While therapeutic applications seem benevolent, they open doors to more troubling possibilities. In 2020, a team at Northwestern University developed a technique using transcranial magnetic stimulation, a TMS, that could enhance specific memories while suppressing others. The researchers found they could effectively strengthen certain memory traces while causing competing memories to fade, effectively prioritizing some experiences over others. Elizabeth Loftus, whose groundbreaking work on false memories began in the 1970s, warns, memory manipulation technologies are developing faster than our ethical frameworks to govern them. We're approaching a point where the distinction between historical truth and subjective experience may become meaningless for the individual. Perhaps most disturbing is research into memory transfer, the direct copying from one memory from one brain to another. In 2022, researchers at Caltech successfully transferred a simple memory between two marine snails using RNA injection. Though primitive, this experiment suggests that memories may have a physical, transferable substrate. Lead researcher Dr. David Glanzman noted, "We've shown that memory is, at its core, a modification of proteins and RNA. If these can be extracted and transferred, there's no theoretical reason why more complex memories couldn't someday be moved between individuals." The military applications have received substantial funding despite minimal public awareness. DARPA's Restoring Active Memory program, launched with $40 million in 2013, initially focused on helping soldiers with traumatic brain injuries. By 2021, the program had expanded to include research on memory enhancement for intelligence operations and interrogation resistance training. A classified offshoot, according to documents obtained through Freedom of Information requests, explores memory resilience against hostile neural intervention. Tacit acknowledgement that memory manipulation could become a weapon. The pharmaceutical industry has also recognized the potential. In 2023, a major pharmaceutical company filed patents for a compound labeled only as XMS224, described as capable of temporary episodic memory suppression. While marketed for treating anxiety disorders, the compound's ability to create discrete periods of anterograde amnesia has raised concerns about potential misuse in everything from date, rape scenarios to corporate espionage. The ethical implications extend beyond individual rights. Collective memory, how societies remember their history, could become subject to manipulation. Imagine targeted memory modification deployed across populations to alter perceptions of historical events or political figures. Dr. Charles Marmer, chair of psychiatry at NYU Langone Medical Center, observes, memory shapes identity, both personal and cultural. Technologies that can reshape memory, therefore, have the potential to reshape who we are as individuals and as a society. Legal systems worldwide remain unprepared for these advances. In 2024, the first court case involving a memory manipulation defense reached the Supreme Court of California. The defendant claimed that memories of his confession had been implanted during interrogation using subtle neurolinguistic techniques. Though the court rejected this defense, Justice Alina Cardenas noted in her opinion, "Our legal system presupposes the reliability of memory and testimony. Advances in neuroscience increasingly challenge this fundamental assumption." The consumer applications have already begun to emerge. Several Silicon Valley startups now offer memory optimization services, using non-invasive brain stimulation to supposedly enhance retention of educational material or professional skills. One company, Neurocall, advertises, "Shape your mind, shape your future," suggesting memory is just another resource to be optimized for competitive advantage. What makes memory manipulation particularly insidious is that its victims may never know what they've lost or gained. Unlike other forms of intervention, successful memory alteration erases awareness of the change itself. As Dr. Stephen Hyman, former director of the National Institute of Mental Health, puts it, "The unique horror of memory manipulation is that it could rob us of ourselves without our knowledge. We would still feel autonomous, or potentially acting on memories and motivations implanted by others." Perhaps most profound is how memory manipulation challenges our concept of personal identity. If our memories can be altered, implanted, or erased, what remains of the self? Philosopher Anil Seth of the University of Sussex argues, "We are, in a very real sense, the stories we tell ourselves about our past. If these stories can be rewritten by external forces, we face not just a technological challenge but an existential one. As we venture further into this territory, we confront the possibility that the most private sanctuary of the self, our memories, may no longer be inviolable. This may prove to be not just one of humanity's most terrifying scientific discoveries, but one that redefines what it means to be human." Mind uploading. What defines you? Your body, your brain, your memories, your consciousness. For millennia, philosophers have debated the nature of identity. But now neuroscientists and computer engineers are forcing this question out of the realm of theory and into laboratories. Mind uploading, the hypothetical transfer of a human consciousness into a computational substrate, represents perhaps the most profound technological possibility ever conceived, one that could redefine mortality itself. The scientific pursuit began earnestly in 2011, when computational neuroscientist Henry Markram launched the Human Brain Project with an ambitious goal: create a complete functional model of the human brain within a decade. While the project has since tempered its timeline, it demonstrated the serious scientific interest in mapping the brain's full connectome, the comprehensive wiring diagram of neural connections that many believe holds the key to consciousness. In 2016, tech billionaire Sam Altman helped launch a startup called Nectum, which developed a preservation technique that maintains brain structures down to the nanometer level. The process, called aldehyde stabilized cryopreservation, won a Brain Preservation Foundation prize for preserving a pig's brain so perfect that every synapse remained intact. The catch: the process is 100% fatal, requiring the brain to be preserved at the moment of death, or potentially before. Nectum's founder, Robert McIntyre, starkly explained the value proposition. "What if we told you we could back up your mind?" The company briefly accepted $10,000 deposits from people on a waiting list before public backlash forced a retreat. Yet the incident revealed something profound: people are willing to die for the mere possibility of digital immortality. Dr. Kenneth Hayworth, president of the Brain Preservation Foundation, argues that mind uploading is a logical extension of the trajectory of neural prosthetics. Indeed, we've already begun the merger of mind and machine. In 2023, Neuralink conducted its first human trials of brain-computer interfaces, allowing paralyzed patients to control computers with their thoughts. The company's founder, Elon Musk, has explicitly stated that neural lace technology represents an early step toward some sort of AI symbiosis where you have an AI extension of yourself. The technical challenges remain immense. The human brain contains approximately 86 billion neurons with 100 trillion synapses, each potentially relevant to our conscious experience. Dr. Sebastian Sung, Princeton neuroscientist and author of Connectum, notes, "If we are our connectum, the technical requirements for mind uploading become clear: capture every connection in a human brain and simulate its function." As of 2024, scientists at Harvard's Connectomics laboratory can map about 1 cubic millimeter of brain tissue per year. At that rate, mapping an entire human brain would take thousands of years. Yet computing power and imaging technology continue to advance exponentially. What's often overlooked in these discussions is the philosophical problem at the core of mind uploading: the continuity of consciousness. Let's say we create a perfect digital copy of your brain. Would you experience being that copy, or would it be a distinct entity that merely thinks it's you? Philosopher David Chalmers calls this the hard problem of uploading. Even if we perfectly capture every neural pattern, there's no scientific way to prove that consciousness transfers rather than just being duplicated. Dr. Susan Schneider, NASA's chair of astrobiology and technical consultant for the film Transcendence, puts it bluntly: mind uploading may be a form of suicide rather than immortality. The biological you dies and something else believes it's you. The social implications are equally staggering. In 2022, economists at the Oxford Martin School modeled a future where wealthy individuals could upload their minds to robotic bodies or virtual realities, potentially living for centuries while accumulating ever more wealth and influence. Their conclusion was stark. Mind uploading could create the most extreme form of inequality ever witnessed, a divide not just of wealth but of mortality itself. The potential for psychological harm is equally troubling. Dr. Rebecca Roach, philosopher at Royal Holloway University, has examined the implications of digital minds experiencing subjective time differently than biological ones. A digital consciousness could experience a subjective millennium in what we perceive as minutes, she notes. This creates the horrifying possibility of near eternal psychological torture. Odd defense applications have not gone unnoticed. A 2024 DARPA white paper explored the potential for cognitive warfare using uploaded partial consciousness. The concept involves creating digital copies of intelligence operatives, expertise without their full personalities, essentially weaponizing fragments of human minds. The document acknowledges the significant ethical complications, but nonetheless outlines potential development pathways. Leading AI researcher Demis Hassabis warns of another danger. An uploaded mind could potentially be modified in ways impossible for biological brains, enhancing certain traits, suppressing others. Who controls these modifications controls the very essence of the uploaded individual. Religious institutions have begun grappling with the theological implications. In 2023, the Vatican convened its first conference on mind uploading, with Cardinal Gianfranco Ravasi concluding, "The soul cannot be reduced to information. A digital copy of consciousness, however perfect, would lack the divine spark essential to human identity." Yet many transhumanists embrace this future unconditionally. The World Transhumanist Association, renamed Humanity in 2008, explicitly advocates for morphological freedom, the right to modify one's consciousness, including uploading it to new substrates. Their manifesto states, "Death is a tragedy. It is not something to be embraced or accepted, but something to be fought with all our technological power." Perhaps most unsettling is how mind uploading could fundamentally change what it means to be human. Hannah Critchlow, neuroscientist at Cambridge University, observes, "Oh, mortality shapes everything about us, our art, our relationships, our goals. Indefinite existence as digital entities would transform us into something fundamentally different from human, regardless of how we began. As we approach this technological singularity, we must ask ourselves, is preserving consciousness worth potentially sacrificing our humanity? The promise of eternal life beckons, but as with all Faustian bargains, the fine print matters. In our rush to transcend our biological limitations, we may be racing toward a future where we save our minds but lose our souls. Brain's plasticity. For centuries, scientists believed the adult brain was fixed, a static organ whose structure, once developed, remained unchanged throughout life. Nobel Prize-winning scientist Santiago Ramón y Cajal, the father of modern neuroscience, declared in 1913, "In adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated." This dogma persisted for decades, cementing our view of the brain as an organ of remarkable capability, but limited adaptability. We now know this fundamental assumption was spectacularly wrong. The human brain possesses an extraordinary capacity for rewiring itself, a phenomenon called neuroplasticity. While this revelation has revolutionized our understanding of brain rehabilitation and learning, it has also exposed vulnerabilities that should give us pause. The same mechanisms that allow recovery from stroke can be hijacked to create addiction. The same neural flexibility that enables language acquisition can reinforce destructive behaviors. The first cracks in the static brain theory appeared in the 1960s, when researchers discovered that environmental enrichment could physically alter rat brains. But the true paradigm shift came in 1996, when neuroscientist Edward Taub published his groundbreaking research on constraint-induced movement therapy. Taub worked with stroke patients whose arms were paralyzed, forcing them to use their affected limbs by restraining their functional ones. Contrary to all expectations, patients regained significant mobility as their brains essentially rewired, recruiting new neural regions to take over for damaged areas. The implications were staggering, as neuroscientist Norman Doidge put it in his 2007 book, The Brain That Changes Itself. The damaged brain can often reorganize itself so that when one part fails, another can substitute. This overturned centuries of medical dogma. But this revelation came with a shadow side. In 2011, researchers at the National Institute on Drug Abuse used brain imaging to demonstrate how addiction physically reshapes neural pathways. Dr. Nora Voko, the institute's director, explained, "Addiction literally changes the brain's structure, hijacking the very plasticity that evolved to help us adapt and survive." The brain's reward system, designed to reinforce beneficial behaviors like eating and reproduction, becomes pathologically focused on substances or behaviors that trigger dopamine release. Perhaps even more concerning is the discovery that our digital environment may be exploiting this plasticity in ways we're only beginning to understand. In 2018, researchers at the University of California found that heavy smartphone users showed altered gray matter volume in regions associated with attention and emotional processing. Lead researcher Dr. Xiaowei Chen observed, "The plasticity that allows us to adapt to tools also makes us vulnerable to becoming adapted by them." This vulnerability extends to our information environment as well. A 2020 study from University College London demonstrated how repeated exposure to misinformation created lasting neural pathways that remained resistant to correction, even when subjects were presented with factual information. The researchers termed this phenomenon neurological entrenchment, where the brain's plastic nature paradoxically leads to cognitive rigidity. The implications for psychological manipulation are profound. In 2022, neuroscientists at Stanford University showed how specifically timed and targeted stimuli could create predictable neural restructuring in volunteers. By carefully controlling exposure to images, sounds, and narrative framing, they could strengthen some neural pathways while weakening others, effectively guiding subjects toward predetermined emotional and cognitive responses. Dr. Michael Merzenich, one of the pioneers of neuroplasticity research, warns, the brain is continuously adapting based on what it processes. In a sense, we become what we pay attention to. This makes the battle for attention not just a marketing concern, but a battle for the physical structure of our brains. Military applications of this research have not gone unexplored. In 2019, DARPA launched its Neural Engineering System Design program, aiming to develop implantable neural interfaces that could enhance soldiers' cognitive abilities. While framed as augmentation technology, internal documents obtained through Freedom of Information requests reveal a secondary goal: understanding how to induce targeted neuroplastic changes for psychological resilience engineering. Beyond external manipulation lies an even more fundamental concern. If our brains continuously rewire based on experience, what happens to the concept of stable identity? Neurophilosopher Thomas Metzinger argues, the discovery of radical neuroplasticity challenges our intuitive sense of being a consistent self over time. Physically speaking, the brain that learned your childhood memories is not the same brain that recalls them decades later. This has profound implications for everything from criminal justice to personal relationships. If we are, in effect, constantly becoming different people as our brains rewire, what does this mean for long-term commitments or for responsibility for past actions? The clinical frontier of neuroplasticity research presents its own ethical quandaries. In 2021, researchers at the University of California, San Francisco successfully used brain-computer interfaces to treat severe depression by identifying and modulating neural circuits involved in mood regulation. While the results were promising, lead researcher Dr. Catherine Skangos acknowledged the double-edged nature of the advance. The same techniques that can heal disordered brain circuits could potentially be used to manipulate healthy ones. Perhaps most unsettling is how this knowledge impacts our relationship with free will. If our decisions emerge from neural pathways shaped by past experiences, and those pathways can be deliberately manipulated, what remains of autonomous choice? Neuroscientist Joyce Hannah Critchlow of Cambridge University notes, neuroplasticity research suggests that free will may exist less as an absolute capacity and more as a continuum influenced by how our neural architecture has been shaped. This doesn't necessarily reduce us to mere automatons. Rather, it suggests that freedom might depend partly on understanding and actively directing our brain's plastic nature. As Dr. Jeffrey Schwartz, pioneer in OCD treatment through directed neuroplasticity, observes, the power to direct attention is perhaps the closest thing to free will we possess. By choosing what we focus on, we literally change our brain's physical structure. The discovery of neuroplasticity represents both promise and peril: the promise of healing, growth, and adaptation throughout life, and the peril of manipulation, addiction, and identity dissolution. As we peer deeper into the brain's remarkable capacity for change, we confront not just a scientific revelation, but an existential one. The physical substrate of who we are is neither fixed nor immutable, but constantly in flux, responsive to both our choices and forces beyond our control. Cellular Senescence, the zombie cells driving our demise, within your body at this very moment. Millions of cells are entering a state that scientists have come to describe with a term straight from horror fiction: zombie cells. Neither fully alive nor completely dead, these cells linger in a limbo-like state, refusing to divide yet stubbornly refusing to die. This phenomenon, cellular senescence, has emerged as one of modern biology's most fascinating and frightening discoveries, reshaping our understanding of aging, disease, and the limits of human lifespan. First identified in 1961 by Leonard Hayflick, cellular senescence was initially observed as a simple limitation. Human cells cultured in a laboratory would divide approximately 50 times before entering a permanent state of growth arrest. This Hayflick limit established a fundamental boundary to cellular immortality and suggested an inescapable biological clock ticking toward our inevitable decline. but the full implications of senescence remained obscure until the early 2000s, when researchers discovered something unexpected. Senescent cells don't simply stop dividing, they transform into metabolically hyperactive entities that secrete a complex cocktail of inflammatory molecules, proteases, and growth factors collectively known as the senescence associated secretory phenotype, or SACE-P. Dr. Judith Campisi, a pioneer in senescence research at the Buck Institute for Research on Aging, describes the process in stark terms. Senescent cells become biological terrorists. They may comprise only a few % of cells in aged tissues, but they can destroy an entire neighborhood through their toxic secretions. The SACE creates a molecular environment that damages surrounding tissues, promotes inflammation, and can even transform healthy neighboring cells into fellow zombies. This cascade effect explains how relatively small numbers of senescent cells can cause disproportionate damage throughout the body. Perhaps most alarming, this process accelerates with age, creating a relentless downward spiral of cellular dysfunction. In 2011, researchers at the Mayo Clinic conducted a landmark experiment, selectively eliminating senescent cells from genetically modified mice. The results were nothing short of astonishing. The mice didn't just age more slowly, they experienced actual age reversal, with improvements in cardiovascular function, bone density, and metabolic health. Their lifespans increased by 25%, and more importantly, their health span, the period of life free from disease, expanded dramatically. Jan van Dersen, who led the Mayo Clinic study, noted, "We were shocked. We expected modest improvements in specific tissues, but instead observed systemic rejuvenation. The implications for human aging are profound." What makes cellular senescence particularly disturbing is its dual nature. Evolution didn't design this mechanism to kill us, quite the opposite. Senescence serves as a crucial anti-cancer mechanism, preventing potentially cancerous cells from dividing by locking them into a non-replicative state. It also aids in wound healing and embryonic development. The protective system that safeguards us in youth becomes the very thing that destroys us with age. This evolutionary trade-off, protection in youth at the cost of deterioration in age, epitomizes what scientists call antagonistic pleiotropy. As evolutionary biologist George Williams theorized, traits that increase reproductive success early in life will be favored by natural selection, even if they cause harm later, after reproduction has occurred. We are, in essence, designed to fall apart. The triggers for senescence read like a catalogue of modern health threats: radiation, chemical toxins, viral infections, excessive caloric intake, psychological stress, and even the air pollution endemic to urban environments can all push cells into senescence. A 2023 study from the University of California revealed that exposure to common air pollutants accelerated senescence in lung cells, potentially explaining the link between pollution and accelerated aging. Perhaps most worrying is the finding that senescent cells actively resist apoptosis, the programmed cell death that normally eliminates damaged cells. They achieve this through upregulating anti-apoptotic genes, essentially deploying molecular shields against the body's own cleanup mechanisms. Peter de Keyser of the University Medical Center Utrecht describes it as cellular narcissism. These cells prioritize their own survival at the expense of the organism. The connection between senescence and chronic disease extends far beyond aging. Researchers have found senescent cells at the core of conditions including Alzheimer's, diabetes, osteoarthritis, pulmonary fibrosis, and atherosclerosis. A 2020 study in Nature demonstrated that senescent cells in the brain drive neuroinflammation and cognitive decline, suggesting that the brain fog experienced by many COVID-19 survivors might result from virus-induced cellular senescence. This discovery has launched a gold rush in pharmaceutical development. Senolytics, drugs designed to selectively eliminate senescent cells, have shown remarkable results in animal models. Compounds like dasatinib and quercetin can clear senescent cells from multiple tissues, reversing age, related pathologies, and extending lifespan. The first human trials began in 2018, focusing initially on specific conditions like diabetic kidney disease and idiopathic pulmonary fibrosis, rather than aging itself primarily for regulatory reasons. James Kirkland, director of the Robert and Arlene Kogod Center on Aging at Mayo Clinic, explains, "We're not treating aging as a disease yet, though it's the major risk factor for most chronic diseases. Instead, we're targeting individual age-related conditions while gathering data on broader systemic effects." The economic stakes are enormous. The global market for senolytics is projected to reach $44 billion by 2030, according to a 2024 analysis by Morgan Stanley. Major pharmaceutical companies have established dedicated senescence research divisions, while biotechnology startups focused exclusively on senolytic development have received billions in venture capital funding. Yet significant questions remain. Senescent cells serve essential functions in wound healing, embryonic development, and cancer prevention. Eliminating them entirely could potentially cause more harm than good. Manuel Serrano of the Institute for Research in Biomedicine Barcelona warns, "We need precision senolytics that can distinguish between beneficial senescence and pathological senescence. The current approach risks eliminating cells we actually need." Some researchers have raised concerns about potential unintended consequences. Lorna Harries of the University of Exeter cautions, "We're intervening in a fundamental cellular process without fully understanding its role across different tissues and life stages. It's like removing a species from an ecosystem. The downstream effects could be unpredictable." Despite these concerns, the potential of targeting cellular senescence remains too promising to ignore. If the results from animal studies translate to humans, senolytics could represent the most significant advance in medicine since antibiotics, not just adding years to life, but restoring vitality to previously inevitable decline. As we unravel the mechanisms of these zombie cells, we confront a profound revelation. The seeds of our destruction may lie not in some external threat, but in the very cellular processes evolved to protect us. Our bodies harbor both the poison and, potentially, the antidote, a discovery both terrifying in its implications and hopeful in its possibilities. Limits of organ regeneration. Slice a salamander's limb, and something miraculous happens. It regrows, complete with bones, muscles, and blood vessels. Cut a planarian flatworm into pieces, and each fragment regenerates into a complete organism. Yet sever a human limb, and the best modern medicine can offer is a prosthetic replacement. This stark contrast represents one of biology's most profound mysteries: why some creatures possess extraordinary regenerative powers, while humans, for all our evolutionary complexity, largely lack this ability. Humans stand at a curious halfway point in nature's regenerative spectrum. Our liver can regrow up to 70% of its mass when damaged. Skin continuously renews itself. Bone knits back together after fractures, but attempt to regenerate a heart, a spinal cord, or a limb, and we hit an insurmountable biological wall, a limitation with devastating consequences for millions suffering from organ failure, spinal injuries, and amputations. The science of regenerative medicine emerged from a simple, haunting question: why not us? Why can a zebrafish regrow its heart while ours forms permanent scar tissue? Why can an axolotl regenerate its spinal cord while human spinal injuries result in permanent paralysis? The answer, scientists have discovered, lies not in what we're missing, but in what we've sacrificed. In 2019, researchers at Harvard's Wyss Institute made a stunning discovery. Analyzing gene expression across regenerative and non-regenerative species, they identified a patent species with limited regenerative capacity don't lack the necessary genetic machinery. They actively suppress it. Dr. Jessica Whited, who led the study, explains, humans have the genetic programs for regeneration. The genes are there, but they're locked away, deliberately silenced by regulatory mechanisms. This evolutionary trade-off likely occurred as our immune systems became more sophisticated. The intense inflammatory response that helps humans fight infection, and cancer also inhibits the cellular plasticity necessary for regeneration. Our biological defenses against disease became barriers to healing. Dr. Kinpos, director of the Regeneration Next initiative at Duke University, frames it starkly. We've essentially traded perfect healing for longevity and cancer resistance. It's an evolutionary bargain that's kept us alive longer but at the cost of complete regeneration. This fundamental limitation has spawned an entire field dedicated to circumventing our biological constraints. Traditional organ transplantation, miraculous as it seemed in the 20th century, faces chronic shortages, with over 100,000 Americans currently waiting for organs and approximately 17 people dying daily while waiting. This crisis has accelerated alternative approaches, each revealing both the promise and the limits of our regenerative potential. Bioengineered organs, grown from a patient's own cells on decellularized scaffolds, represent one frontier. In 2018, researchers at the University of Texas Medical Branch successfully engineered functioning lungs using this approach, only to face a harsh reality. The organs failed shortly after transplantation into animal models. The intricate vascular network necessary for long-term function proved impossible to replicate using current techniques. Dr. Laura Nicholson, founder of Humacyte and pioneer in engineered blood vessels, acknowledges the challenge. We're discovering that an organ isn't just an assembly of cells, but a dynamic, self-regulating ecosystem that develops through complex embryological processes we still don't fully understand. Replicating this complexity has proven far more difficult than anticipated. The limitations extend to stem cell therapies, once heralded as regenerative medicine's holy grail. While successful in treating blood disorders and some immunological conditions, stem cells haven't delivered the broad regenerative capabilities initially promised. Injected stem cells often die before integrating into tissues, fail to differentiate appropriately, or trigger immune rejection. Perhaps most concerning is the discovery that many stem cell populations deplete with age, precisely when regenerative needs increase. A 2021 study from Stanford University found that neural stem cells, critical for brain repair, decrease by approximately 70% between ages 20 and 70. This decline creates a cruel paradox. Our regenerative capacity diminishes as our bodies accumulate damage. The field of 3D bioprinting has encountered similar biological boundaries. While researchers can print organ-like structures with living cells, these constructs lack the microvascular networks necessary for nutrient delivery and waste removal beyond a few millimeters of tissue. Without blood vessels, printed organs quickly develop necrotic cores. The vascularization problem represents a fundamental limit we haven't overcome, explains Dr. Anthony Atolla, director of the Wake Forest Institute for Regenerative Medicine. We can print simple tissues, but complex organs need thousands of microscopic vessels that must connect seamlessly to the patient's circulatory system, a challenge that has thus far proven insurmountable. These biological constraints have forced a reckoning within the field. Initial promises of growing replacement organs on demand have given way to more modest goals, focused on enhancing our limited natural regenerative processes, rather than recreating complete organs. One promising approach involves temporary reactivation of developmental pathways that normally shut down after embryonic development. In 2023, researchers at the Salk Institute used a combination of 4 transcription factors to temporarily reactivate embryonic gene expression in mature cells. This induced a transient state of cellular plasticity that enhanced healing in mouse models of heart injury. Rather than fighting our evolutionary inheritance, we're working with it, explains Dr. Juan Carlos Pizarro-Belmont, who led the research. By temporarily unlocking developmental programs, we can enhance regeneration without disrupting the immune surveillance that protects us from cancer. Yet even these approaches face significant safety concerns. The same cellular plasticity that enables regeneration can lead to tumor formation if not precisely controlled. Several clinical trials using regenerative approaches have been halted after participants developed cancerous or pre-cancerous growths. The philosophical implications of these biological limits extend beyond medicine. Our inability to regenerate perfectly has shaped human culture, religion, and society. From ancient healing rituals to modern prosthetics, humans have developed technological and cultural adaptations to compensate for what our biology cannot provide. Dr. Lino Hooks, bioethicist at the University of Edinburgh, notes, "There's a profound irony in our quest for perfect regeneration. Our technological creativity, our prosthetics, our medicines, our scientific endeavors emerged partly because we cannot naturally regenerate. Our biological limitations have driven our cultural evolution. As we push against the boundaries of our regenerative capacity, we confront not just technical challenges but existential questions about human mortality and identity. Perhaps the most terrifying scientific discovery isn't that perfect regeneration exists in nature, but that it remains just beyond our grasp, close enough to glimpse, yet separated by biological barriers we've only begun to understand. Geoengineering Proposals. Imagine a fleet of specialized aircraft releasing millions of tons of sulfur dioxide into the stratosphere, creating a thin veil of particles that reflects sunlight back into space. Picture vast mechanical forests of artificial trees, extracting carbon dioxide from the atmosphere at rates thousands of times more efficient than their natural counterparts. Envision fleets of autonomous ships spraying seawater into clouds, brightening them to reflect more sunlight. These aren't science fiction scenarios, but real proposals from respected scientists to address climate change through deliberate, large-scale intervention in Earth's natural systems, a practice known as geoengineering. As global temperatures continue to rise despite decades of climate negotiations, these radical interventions have moved from theoretical curiosities to serious considerations. Yet they represent perhaps the most profound gamble in human history, attempts to manipulate planetary systems we barely understand, with consequences that could affect billions of lives across generations. The most controversial geoengineering approach, solar radiation management, SRM, aims to reflect a small percentage of incoming sunlight back into space. The concept draws inspiration from volcanic eruptions. When Mount Pinatubo erupted in 1991, it injected 20 million tons of sulfur dioxide into the stratosphere, cooling Earth by about 0.55 UC for nearly 2 years. Proponents suggest we could replicate this effect, deploying specially designed aircraft to continuously release sulfate aerosols into the upper atmosphere. In 2022, researchers at Harvard University's Solar Geoengineering Research Program conducted preliminary atmospheric tests, launching small balloons to release calcium carbonate particles in the stratosphere. While merely testing equipment and measuring dispersal patterns, the experiment unleashed international controversy. Critics argued it crossed a dangerous threshold, beginning experimentation with a technology that might seem benign in small doses but carries catastrophic risks at scale. Dr. Alan Robock, a climate scientist at Rutgers University, who has identified 27 potential risks of stratospheric aerosol injection, warns, "We're talking about deliberately changing the climate of the entire planet. The potential for unintended consequences is enormous. Once we start, we're committed potentially for centuries. Those unintended consequences could be devastating." Models suggest SRM could disrupt monsoon patterns that billions of people depend on for agriculture, potentially causing widespread famine. It could deplete the ozone layer, increasing skin cancer rates globally. Perhaps most disturbingly, it creates what scientists call termination shock. If the aerosol program were ever halted, temperatures would rapidly spike, potentially rising several degrees within a decade, far faster than ecosystems or human societies could adapt. Catherine Richardson of the University of Copenhagen frames it starkly. Stratospheric aerosol injection is like taking aspirin for climate change without addressing the underlying disease. It might temporarily mask symptoms while allowing the fundamental problem, greenhouse gas concentrations, to worsen. are carbon dioxide removals, CDR. The second major category of geoengineering aims to extract CO directly from the atmosphere. While seemingly less risky than solar engineering, CDR approaches face their own troubling limitations. Direct air capture technologies currently consume enormous energy. One analysis suggests capturing just 10% of current annual emissions would require approximately 25% of global energy production. Bioenergy with carbon capture and storage, a BCCs, another widely discussed approach, would require converting vast land areas to energy crops, potentially threatening food security and biodiversity. A 2023 analysis in Nature Sustainability calculated that removing enough carbon to meet climate targets through BCCs alone would require land equivalent to approximately 1/3 of current global cropland. Ocean fertilization, seeding vast ocean regions with iron to stimulate plankton blooms that absorb carbon, has shown disappointing results in field trials while raising concerns about marine ecosystem disruption. A 2012 experiment off Canada's west coast sparked international outrage when a private company conducted unauthorized iron dumping, highlighting the potential for rogue actors to implement geoengineering without international consent. This governance challenge may be geoengineering's most terrifying aspect. Who decides whether to deploy technologies affecting the entire planet? Who controls the global thermostat? A 2024 report from the Carnegie Climate Governance Initiative warned of a governance gap, where the nations most capable of deploying geoengineering, primarily wealthy, technologically advanced countries, are not those most vulnerable to its side effects. Dr. Sheila Jesanoff, professor of science and technology studies at Harvard Kennedy School, notes, geoengineering creates unprecedented power asymmetries. A handful of nations could unilaterally alter the climate for everyone, effectively imposing their risk preferences on billions of people who had no voice in the decision. This power dynamic becomes especially concerning given military interest in climate intervention. The U.S. Defense Advanced Research Projects Agency, a DIRPA, has funded several geoengineering research initiatives, raising concerns about dual-use technologies that could weaponize weather modification. A 2020 RAND Corporation report acknowledged that stratospheric aerosol technology could potentially be deployed to create localized climate effects for strategic advantage. Even if governance challenges could be resolved, geoengineering proposals suffer from what philosophers call the moral hazard problem, the risk that pursuing technological fixes reduces incentives to address root causes. As Oxford philosopher Henry Shue puts it, geoengineering offers powerful interests a narrative of salvation through technology, potentially undermining the political will for the systemic changes needed to address climate change. The implications extend beyond environmental concerns to questions of intergenerational ethics. Once deployed, many geoengineering approaches would require continuous maintenance for decades or centuries. Solar radiation management, in particular, creates obligations for future generations who had no say in the initial decision. Stop the program, and temperatures spike catastrophically. Continue it, and future societies remain dependent on increasingly aging infrastructure to maintain a livable climate. Harvard philosopher Stephen Gardner calls this the perfect moral storm. Decisions made today creating binding obligations for generations unborn, with no mechanism for their consent. We're essentially colonizing the future, he argues, imposing risks on people who cannot possibly have a voice in today's decisions. Perhaps most troubling is how geoengineering might transform humanity's relationship with nature. For millennia, humans have adapted to Earth systems. Geoengineering reverses this relationship, attempting to make Earth adapt to human needs. This represents what environmental philosopher Clive Hamilton calls playing God. Assuming control over planetary systems evolved over billions of years, whose complexity we still barely comprehend. As we contemplate deliberate intervention in Earth's climate system, we confront not just technological challenges, but profound questions about human wisdom and foresight. The most terrifying aspect of geoengineering may not be any specific risk, but the hubris of believing we can predict and control the consequences of planetary scale manipulation, when our brief experiment with unintentional climate modification has already produced so many unforeseen impacts. The potential of fusion energy. Nuclear fusion stands as potentially humanity's most transformative energy discovery. The same process that powers our sun and stars could revolutionize how we generate electricity on Earth. Unlike conventional energy sources with their limited reserves and environmental impacts, fusion promises something unprecedented, nearly limitless clean energy from abundant resources. At its core, fusion energy harnesses the fundamental forces of nature. When light atomic nuclei, typically isotopes of hydrogen like deuterium and tritium, are forced together under extreme conditions, they fuse to form heavier elements, releasing enormous amounts of energy in the process. A single gram of fusion fuel could theoretically produce the same energy as 10 tons of coal. What makes fusion particularly remarkable is its fuel source. Deuterium can be extracted from ordinary seawater. About one water molecule in 64020 contains deuterium. This means our oceans hold enough fusion fuel to meet global energy needs for millions of years. Tritium, though rare naturally, can be produced within the fusion reactor itself from lithium. The potential environmental benefits are equally staggering. Fusion produces no direct carbon emissions or air pollution. It generates no long-lived radioactive waste. Unlike conventional nuclear fission, the reaction products are primarily helium, an inert, non-toxic gas, and neutrons that can be captured for their energy. Recent breakthroughs have brought fusion closer to reality. 2022, scientists at the National Ignition Facility achieved fusion ignition for the first time in history, producing more energy from fusion than the laser energy used to drive it. This watershed moment proved that net energy gain from fusion is physically possible, something that had eluded researchers for over 60 years. Meanwhile, the International ITER project in southern France represents the most ambitious fusion experiment ever attempted. This tokamak-type reactor aims to produce 500 megawatts of fusion power from 50 megawatts of input power, sustaining the reaction for extended periods. Though still under construction, ITER will demonstrate the feasibility of fusion as a large-scale energy source. Private investment has accelerated fusion development dramatically. Companies like Commonwealth Fusion Systems, a MIT spin-off, TI Technologies, and General Fusion have attracted billions in funding. Commonwealth Fusion Systems' high-temperature superconducting magnets have broken field strength records, potentially enabling smaller, more economical fusion devices than previously thought possible. The safety profile of fusion adds to its appeal. Unlike nuclear fission, fusion reactions cannot sustain a chain reaction. They require precise conditions to occur. If anything disrupts these conditions, the reaction simply stops. There's no risk of meltdown or runaway reactions. The total amount of fuel in a fusion reactor at any moment is minimal, mere grams, further enhancing safety. The strategic implications of mastering fusion energy would be profound. Nations could achieve energy independence without relying on fossil fuel imports. Energy-intensive industries could operate without environmental penalties. Remote regions could establish self-sufficient power generation without massive infrastructure. Fusion could also enable other technologies previously considered impractical due to energy constraints. Desalination at massive scale could address global water shortages. Energy-intensive carbon capture could mitigate climate change effects. Even space exploration would benefit from the high energy density fusion provides. The scientific knowledge gained from fusion research extends far beyond energy production. Plasma physics advances have applications in materials science, medical technologies, and understanding astrophysical phenomena. The computational models developed for fusion simulations have improved scientific computing across disciplines. Challenges certainly remain. Plasma containment at 150 million degrees Celsius are hotter than the sun's core, presents extraordinary engineering difficulties. The neutron flux degrades reactor materials over time. Economic viability requires extending component lifetimes and improving reliability. Yet each of these challenges has seen significant progress in recent years. The timeline for commercial fusion has compressed dramatically. While fusion was once joked about as always 30 years away, multiple companies now project demonstration plants operating in the 2030s. This accelerated timeline reflects both technological breakthroughs and increased investment. Unlike many scientific pursuits, fusion energy development has fostered remarkable international cooperation. The ITER project involves 35 nations working together, including sometimes adversarial powers like the United States, Russia, and China, highlighting fusion's potential as a global solution transcending political differences. Perhaps most profoundly, fusion represents humanity's first real opportunity to move beyond resource scarcity in energy production. Throughout history, civilizations have been constrained by limited energy supplies. Fusion could fundamentally alter this equation, providing abundant energy without environmental degradation. As fusion transitions from scientific possibility to industrial reality, it may well represent our most significant technological achievement, harnessing the very process that lights the stars to secure humanity's energy future. Genetic mutations from pollution. In the quiet dance of our DNA lies a delicate code, billions of years in the making. But in recent decades, we've discovered something profoundly disturbing. This ancient blueprint, the foundation of all human life, is being silently altered by the very world we've built around us. Industrial chemicals, air pollutants, and synthetic compounds are rewriting our genetic future in ways we're only beginning to comprehend. This isn't science fiction. It's the troubling reality emerging from laboratories worldwide. Consider the inhabitants of the Love Canalnade in Niagara Falls. In the 1970s, residents discovered their homes were built atop 21,000 tons of toxic waste. What followed was a disturbing pattern of birth defects, miscarriages, and genetic abnormalities. DNA analysis later revealed specific mutations in exposed populations. Tangible evidence of how industrial chemicals had permanently altered their genetic code. More alarming is what scientists call transgenerational epigenetic inheritance. Research by Dr. Michael Skinner at Washington State University demonstrated that when pregnant rats were exposed to common industrial chemicals, the genetic damage appeared not just in their offspring, but in generations that followed, up to the great-grandchildren who had never directly encountered the toxins. The implications for humans are chilling. Pollution today could be programming genetic abnormalities for generations yet unborn. The mechanisms are increasingly understood. Polycyclic aromatic hydrocarbons, SPH from vehicle exhaust and industrial emissions can directly bind to DNA, creating what scientists call HODUCs that distort the double helix structure. A 2019 study in the journal Environmental Science, Nanotechnology found that children in heavily polluted urban areas had significantly higher levels of these DNA adducts than those in cleaner regions, with corresponding increases in mutation rates. Heavy metals present another insidious threat. A landmark study tracking mercury exposure in Minamata, Japan, where industrial waste contaminated local seafood, found distinctive chromosomal abnormalities persisting decades after exposure ended. These mutations correlated with neurological disorders that appeared even in descendants born years after pollution controls were implemented. Perhaps most concerning are endocrine disrupting chemicals, as EDCs found in everything from plastics to pesticides. These compounds mimic hormones, interfering with the epigenetic markers that determine which genes are expressed. The Rotterdam study, following over 10,000 participants, found that higher blood levels of EDCs, like bisphenol, are correlated with measurable alterations in genes regulating metabolism and immune function. These changes weren't just temporary. They became permanently embedded in the genetic code. Radioactive pollution presents its own terrifying genetic legacy. Following the Chernobyl disaster, researchers documented a 600% increase in certain genetic mutations among children born in contaminated regions. The Genome Research Institute's 30-year follow-up study found these mutations weren't random. They clustered in specific DNA repair genes, potentially increasing cancer susceptibility for generations. Air pollution's genetic impact extends beyond obvious industrial areas. A 2021 Harvard study examining urban particulate matter exposure found it increased mutation rates in sperm-producing cells, potentially transmitting genetic damage before conception even occurs. Meanwhile, the Barcelona Institute for Global Health demonstrated that pregnant women exposed to high levels of nitrogen dioxide birthed babies with measurable alterations in genes regulating immune response. Agricultural chemicals contribute to this genetic burden. The Agricultural Health Study, following over 89,000 farmers and their families, found elevated rates of specific genetic variations in populations exposed to common pesticides. These mutations appeared in genes controlling cellular division and DNA repair, fundamental processes whose disruption can lead to cancer and developmental disorders. What makes these findings particularly disturbing is the concept of the mutation load. Each genetic alteration may seem insignificant in isolation, but collectively, they can overwhelm our DNA's natural repair mechanisms. The Columbia Center for Children's Environmental Health has documented steadily increasing mutation rates in urban populations over the past 50 years, correlating directly with industrial expansion. This cumulative burden threatens to undermine human genetic integrity on a timeline far shorter than natural evolution. Even more troubling is our growing understanding of genetic hotspots, DNA regions particularly vulnerable to environmental damage. Research from the Environmental Genome Project has identified sequences that mutate at up to 100 times the normal rate when exposed to common pollutants. Many of these regions control crucial developmental pathways, explaining why pollution exposure during pregnancy poses such severe risks. The geographic distribution of these mutations reveals an environmental justice crisis. The Cancer Alley region along the Mississippi River, home to over 150 industrial facilities, shows mutation rates up to 7 times the national average among predominantly low-income minority communities. Similar patterns appear worldwide, with genetic damage concentrated in populations with the least political power to address industrial pollution. Despite these alarming findings, most environmental regulations fail to consider genetic impacts. The vast majority of the 80,000 chemicals in commercial use were approved before modern genetic testing, and manufacturers aren't required to evaluate their products' mutagenic potential. Even known mutagens like benzene and formaldehyde remain in widespread use, their genetic legacy quietly accumulating in exposed populations. As our detection methods become more sophisticated, we're discovering that genetic damage occurs at pollution levels far below current, safe thresholds. A 2022 study in Environmental Health Perspectives found measurable DNA damage in children exposed to air pollution at just 50% of EPA standards, suggesting our regulatory framework remains dangerously inadequate. The terrifying reality is that we've conducted a massive, uncontrolled genetic experiment on ourselves. Each industrial revolution has introduced new chemicals without understanding their genetic consequences. Now, as we decipher the results, we face the sobering possibility that we've altered the very foundation of human biology, potentially compromising the genetic heritage passed down since the dawn of our species. This isn't merely about increased cancer rates or developmental disorders. Though these are tragedy enough, it's about a fundamental reshaping of human genetics in ways that could permanently diminish our resilience, intelligence, and health. The most frightening scientific discovery may be that in our rush to transform our environment, we've unwittingly transformed ourselves at the most fundamental level. And these changes cannot be undone.